morning, everyone. Can you find? Yes, here, yes, I hope online too. So welcome back, everyone, to our winter school on safety and beneficial properties of fermented products. So today I'll be the moderator and I will start the talk. The first talk is by myself, microbial interaction. My name is Uelinton Pinto, and I'll just present myself to you. So I hold a Bachelor of Science degree in Food Engineering by the University of Minnesota. Also a Master's degree in Microbiology by the Federal University of Minnesota. I have a PhD in Microbiology from Monteiro University, and I was a professor of Food Science and Technology, Food Microbiology at the Federal University of Ouro Preto from 2011 to 2015. When I came to USP, so I'm a professor at the University of São Paulo since 2015. I spent a year at Harvard as a visiting scholar from 2019 to 2020. And since 2016, I am an associate researcher at the Food Research Center, which is located here at USP. And I have been a research scholar from CRISPR-Cas since 2023. So my talk today will be about microbial interactions in food ecosystems, which is right there. And I'm going to start right now. So please let us know if you have any problems online. Vlad is here. Gabriel is also here to help. So if anything happens, just let us know. So the topic is microbial interactions in food ecosystems, which is like the introduction for the day, because we're going to be talking about a lot of interactions in different systems. So we're going to see throughout the talks that I'm going to be talking like about the major interactions that we can have, but also with a focus on microbial signaling. And then throughout the day, we're going to have people talking about phage and bacteria interactions in dairy products, especially cheese. We're going to have people talking about interactions in cheese by itself, like by the end of the day, among other interactions. So it's, I would say that it's the day that I like a lot because it's my research area. So I really enjoy all the talks, and everybody that was invited is going to be talking about stuff that I really like to hear. And I hope you share the same expectations that I share. So the topics for this talk are microbial interactions in foods, and then I'm going to tackle a little bit on the trophic interactions in which microorganisms exchange metabolites with one another. Then we go on to talk about chemical signaling, especially quoron sensing. If you don't know what that is, I'm going to explain later on. And then we end talking about quoron sensing inhibition. So what are microbial interactions and why are they important? When we talk about microbial ecology, we are talking about all of these things that are here. I don't have a pointer, so I'm going to try to use the mouse. On the audience, you can see. I don't know if online they can. If they don't, let me try one thing here. It's not working. Yeah, I wanted to put the... but for you, like online, they can't. Oh, all right, thanks. So microbial ecology, it's basically microbial composition and function. And the microbial composition and function are affected by abiotic factors and biotic factors and their interactions. So when we talk about abiotic factors, we're talking about intrinsic and extrinsic factors. Everybody that has studied food microbiology has heard about these themes. We talk about a lot on microbial ecology or food microbial ecology. So intrinsic factors, they are related to the food. So you're talking about water activity, pH,
redox potential. So it's what it's inherent to the food itself. In extrinsic factors, it's where the food is stored, let's say. So we're talking about temperature, mostly, which really influences how microorganisms will multiply. And we're talking about the atmosphere. So is it in the presence of oxygen? Is it in the vacuum? Or do we have a mix of other gases that are influencing this microorganism's growth? And then we have the biotic factors. I would say that intrinsic and extrinsic factors influence the most. So if you have, let's say, a product that has a very low pH, that's going to influence a lot who is going to be growing in that product. So you're not going to have the major spoilers like Pseudomonas, for instance, that will not be able to grow in this very acidic environment. If you have a product that has a very low water activity, very few organisms can grow in this product, right? So if you have a product that is under vacuum, so absence of oxygen, so no organisms that need oxygen will grow. So we'll be talking about spoilage or food risk, food safety risk related to botulinum, but not about other organisms because they're not going to be able to grow. But taking that aside, let's say fresh products, we're going to have, of course, the influence of the intrinsic, extrinsic factors, but also we're going to have a lot of interactions between these microbes. And when we are talking about fermentation, which is like a major theme for this course, in fermentations, we have a lot of interactions, especially when we are talking about fermentation of indigenous products. So let's say artisanal cheese is made with raw milk and endogenous culture. So you have a lot of interactions between these microorganisms besides the interactions that happen with the intrinsic and extrinsic factors. So we're going to be unpacking all of this throughout the talk. So let's talk about these biotic factors, which are microbial interactions. And these I can subdivide into two categories. So the first one is trophic interactions, which is basically metabolic exchanges. When we talk about trophic interactions, the major thing that happens is cross-feeding. And in microbiology, I like to say that we usually have microbial succession. So if you have a spoilage or if you have a fermentation, it usually happens with one group of microorganisms, then conditions change, and then you have another group of organisms that will grow, and following that, another group, and so on and so forth. So spoilage happens like that. Some fermentations also happen like that. So I'm going to be showing you in the next few slides a few examples of microbial interactions and cross-feedings. But also, besides cross-feeding, which seems like something positive, right? So you have one organism, and that organism is creating an environment that is helping the next organism to grow. So it's like a positive interaction. But we also have negative interactions. So you have antagonism. So you have one organism that is inhibiting the other organism to grow. And this is particularly true when we talk about bacteriocins. So you have one organism producing this antimicrobial peptide, which will inhibit several other organisms. So this is a competition. Or you have one organism that is producing some kind of acid or ethanol, and this is also inhibiting a series of other organisms. So you have positive interactions and you have negative interactions. And sometimes you have neutral interactions. Sometimes one organism produces one substance that has no effect on the neighbor organism. All of this inside these trophic interactions. And in addition to that, we also have chemical signaling. And this is what we call quorum sensing nowadays. But chemical signaling is basically a way that one bacteria or one organism. Not only bacteria can do that, but usually we talk about bacteria. So it's one way that a bacterium cell can produce a chemical signaling, a cue, that will travel throughout the medium and will activate another bacterium. So basically, it's sending signals, and these signals will tell the population to do something. And most of the talk will focus on this, but first, let's talk about the trophic interactions. This slide appeared yesterday. It always appears in every talk, and I always say that. So because this is like summary of food microbiology or food microbial ecology. So you have a model system here in which you have a cheese or the manufacturing of a cheese. And in the beginning of the manufacturing, you have several factors that will affect microbial growth. So first of all, you have in the beginning of production, I can't really see from here, you have pasteurization, cutting, way draining, salting. All of these influence the microorganisms that are present. Sometimes you don't have pasteurization. Sometimes you just have raw milk. So you have more microbial species present. In the beginning, you have more oxygen. This will also affect who is growing. And you have the influence of the temperature. What is the temperature that this cheese is being made? So this is going to be influencing also. And you have this, in this paper, they're talking about technological processes, but mostly it's pH. In the beginning, the pH of is going to be close to the pH of milk, and then it's going to be acidifying, and the pH is going to drop. And you have concentration of sodium chloride, so most cheeses are salted. Several different kinds of methods to salt, sometimes directly in the milk, sometimes on top of the cheese. And you have water activity. And this is where I spot a mistake, because water activity is not percent, it's from 0 to 1. So just clarifying that not every figure is perfect. This one was published with a mistake. And when we talk about the ecological processes, we have all of these organisms, right? So, and some of them will be lost by ecological drift, by chance. So they are present in the environment, they are present in the milk, but somehow they got lost in the middle of the way and they are not in the product. Some of them may undergo some mutation, so you have diversification. So this organism in blue is changing to this other color. And you have environmental selection. And this is very important because the environment is going to dictate who is going to be present next to do whatever the organism is going to be doing. And so at the beginning, you have mostly because also we add starter cultures, which are lactic acid bacteria. So we have mostly lactic acid bacteria, but we also have enterobacteriation. Even if we use pasteurized milk, they are not completely destroyed. And we have also coagulase negative staphylococcus. Sometimes we have coagulase positive as well, but they are not desired. So these organisms will be interacting. And as the ripening happening, so first you have manufacture, then you have ripening. You keep this cheese in a cave, you keep it in a room at conditions that are controlled. And throughout time, this is the process of ripening. So during ripening, the temperature is controlled, time, humidity, and this is going to influence who is going to come next. So next here we have yeasts. So it's 2 yeasts here as cited. And then as ripening happens and the type of cheese as well, because there are several kinds of cheese. Yesterday we saw in the talk of Ana Barreto that all kinds of cheeses, and depending on the technical procedure you use, you're selecting for different kinds of organisms. But here mostly, in the end, you're going to have actinobacteria and some fungi and lots of coagulase negative staphylococcus. And this is going to be probably mentioned by the last talk they're going to have today by Rachel Dutton, which she uses cheeses as tractable systems for microbial interactions. so these microorganisms will be interacting with one another sometimes, and this we call it biotic factors. So you have competition, you have neutral interactions, and you have cooperation. So in the cooperation, you can have mutualism, synergism, commensalism, basically one organism helping the other, or both helping each other. And you have also competition. And in this competition, we usually like to study lactic acid bacteria. We like to select them because they are good competitors. They inhibit pathogens. They can produce bacteriocins, acids, and other compounds. And in this competition, we like to give them advantage so that they would inhibit our pathogens of control. For instance, Listeria, Staph, and E. coli. So let's talk about, I'm going to try to control my time because I tend to talk too much. Some interactions that happens in several kinds of products. So in yogurt, we have two species, as was talked about yesterday. So one side you have Streptococcus thermophilus, and the other one you have the Lactobacillus bulgaricus. And these organisms will interact with one another, and they will interact with the matrix. In the beginning, it's milk. But as both organisms produce lactic acid, this lactic acid, as well as EPS and other compounds, they will influence the milk and will actually decrease the pH and will coagulate the casein, right? And these organisms will exchange several compounds with one another. And basically what happens is that they're helping each other. So they grow by themselves, isolated by themselves in milk, you can do that. But when they grow together, they grow better. And that's why this works so well to make yogurt. When we talk about Swiss type cheese, we have another classical cross-feeding. First, you have the LAB, which will use mostly the sugars, and will produce lactates. Of course, they're going to be producing antimicrobials, vitamins, exopolysaccharides, aromas, and all of that. But lactates will be used in this microbial succession by the propionic bacteria. And propionic bacteria will use lactate, produce acetate, propionate, and CO2, which will produce those big eyes that we see in Swiss type cheese. So this is a classical microbial interaction, microbial succession, and it's relatively simple when we compare with other systems. For instance, we could talk about microbial succession in artisanal cheese. It's a lot more complex than with Swiss type cheese, which is made with pasteurized milk. Here's just one example about microbial interactions in cocoa fermentation. And it's quite complex, but mostly in the beginning, you have the presence of yeasts and lactic acid bacteria. They grow mostly at the same time, but we usually like to say that yeasts start first. They start using the molecules in an anaerobic phase, and they're going to be using sugars, mostly sucrose, breaking down to glucose and producing alcohol or ethanol. And the lactic acid bacteria will also use sucrose, mostly fructose, glucose, and producing lactic acid bacteria, as well as several other compounds. But the most important compound here, for the purpose of the talk, at least, is lactic acid bacteria. And as this fermentation keeps going, temperature of the mass of the cocoa beans start to increase. And this is a factor that's going to affect the next phase of fermentation. So after a few days, I think it's 3 days, a person comes and stirs the mass of the cocoa beans and the pulp. And this brings aeration to the system. And because there is ethanol, lactic acid, and other compounds, mostly lactic acid, it's a cue for the bacteria that produce acetic acid or acidic acid bacteria. So they're going to use the lactic acid and produce acetic acid. And this makes it's exothermic reaction, so increases the temperature of the mass to up to 50 degrees. Together with ethanol, the acids, and this high temperature, this kills the beans or the germ present in the bean of the cocoa. And with that, you're going to have the release of a lot of compounds which are related to the flavors that we recognize in chocolate. So fermentation has to happen in this way. You know, yeasts, LAB, and then acetic acid bacteria. And what is happening here is basically cross-feeding, right? So one organism is producing a metabolite that is being used by the next. With Elaine, who is also a professor that is part in this work, she found out that there is also quorum sensing or quorum sensing inhibition happening between these organisms. So besides cross-feeding, these organisms are also talking to each other or interfering in the communication of the competitors. And this is going to be what we're going to be talking about now or next. So what is quorum sensing? It's a type of microbial interaction. It's an intercellular type of signaling, and it happens at high cell densities. So quorum sensing, we can just say that it's a cell-to-cell communication mediated by chemical signals according to the population density. So as microorganisms accumulate, they liberate molecules in the environment. These molecules increase in concentration until they reach a threshold. And that threshold tells the population to synchronize, to do something together. So for instance, if you think about the production of an enzyme that will break down some complex substrate, one isolated organism producing one enzyme or a few enzymes not going to be able to produce a lot of substrate for growth. But when lots of organisms are producing these enzymes together, they can break down huge amounts of substrates, which will help them to grow. So this is one way of seeing this. Or if you think about infection, you have one organism alone trying to overcome an organism like us, a complex organism. That organism alone producing a virulence factor may not affect anything. But if that organism can hide from the immune system and reach a population high enough that all of them producing a virulence factor together, then they will have strength or power, actually, to cause something that is going to be really affecting the host. There's a very nice talk by Bonnie Bassler, who's one of the leading scientists in the field. It's a TED Talk, so you can watch that later, How Bacteria Talk. It's really nice. You can watch it. So what is controlled by quorum sensing? Why is it so important? And why so many people are researching this field? There are many phenotypes that are important that are controlled by quorum sensing. For instance, biofilm formation, which affects many areas of microbiology, production of virulence factors, metabolism optimization, conjugation, which is basically exchange of genetic material or exchange of plasmids between bacteria, competence, which is acquisition of DNA from the environment. This is also controlled by quorum sensing. Motility, especially motility over surfaces, which we call swarming. Sporulation, resuscitation from the viable but non-culturable states, among others. So it has implications in very diverse areas of microbiology. So how does the system work? There are several kinds of molecules and differences between one organism and the other, but the canonical system works like this, and this was first discovered in Vibrio fischeri, which is a bioluminescent bacteria that lives in the organ of a squid, which is in Portuguese is some kind of lula, so it's a squid. And inside that organ, which is a luminous organ, this organism grows, and when it reaches high cell density, it produces light. So it's not the squid that produces the light, it is the bacteria inside this organ, all right? So how does the system work? So we have here a locus on the chromosome of the bacterium, and this locus has basically three main components. So one of the components is a gene which codes for a regulator, which in this case it's called LuxR because lux, right, lights. A Lux box in which is recognized by the LuxR protein when the protein is activated, and genes that are regulated by the LuxR protein, which is the regulator. It turns out that the first gene of this operon, it's the enzyme that makes the signal. In this case, the signal is called acylhomoserine lactone. So this signal, which combines an amino acid and a fatty acid, so things that are very common in the cell. So the enzyme combines these two molecules and produces the signal. This signal can diffuse freely from the cytoplasm to the exterior of the cell. And the production of this enzyme, of the activation of this operon, is very low at low cell densities, but it happens at basal level, as we would say. So one cell or the other is producing just a little bit of this enzyme, just a little bit, so that a few molecules are produced and they are liberated in the environment. And if the cell keeps at low cell density, this concentration, it's going to be low. But as cells start to grow, especially when we think about the environment of the organ inside the squid, cells will accumulate, and more cells producing even at basal level will increase the concentration of homoserine lactone, so that at a certain threshold, this concentration will activate the regulator or the activator. So when it binds to the LuxR protein, the protein dimerizes, so it forms a dimer, and this dimer is active, so it binds to the Lux box, which is basically a region in the promoter region of the operon, and it's a signal to produce high, massive amounts of whatever is being controlled by quorum sensing. So LuxR binding here produces a lot of everything that is under its control, so a lot of more LuxI protein. So it's like a positive feedback loop. So more homoserine lactone will be produced, and the whole operon, which codes for the enzymes that make light, luciferases and all those things that are needed to produce light. So all of a sudden you have a culture that it's at high cell density, it's not producing light, and from an instant to the other, boom, everybody is producing light, and then the tube becomes bright. You can see it in the dark, or you can see the colony producing light also. It's possible to see. So this is a picture from a student from the lab, and he took it in the dark room. So it's quite pretty when you see this plate shining like that. So I spent 5 years of my life working with agrobacterium tumefacients. Well, let's go back a little bit. So this is very interesting, right? Okay, this bacteria has this, produces light, so what? This was discovered in the 80s, especially the genetic circuit, the structure of the molecule, the signaling molecule. It wasn't called quorum sensing back then. But then, because people started sequencing genes in bacteria, in the beginning of the 90s, they found out that some other bacteria have genes that were homologous to LuxR and LuxI. So they would see these genes that looked like LuxR and LuxI in bacteria that did not produce light. So they started asking, well, what are these genes doing? We know that it's present in Vibrio fischeri, but why is it present in Pseudomonas aeruginosa, which is a terrible pathogen? Why is it present in Agrobacterium tumefaciens, which is a phytopathogen? And at the beginning of the 90s, people found these genes in these other organisms and started making mutations and see what happened to these cultures. in agrobacterium tumefacients, it was found that the homologous genes to LuxR and LuxI are called TRA-R and TRA-I, and they are important for conjugation and other things, as we found later on. So as I was saying, I worked with agrobacterium tumefacients during my PhD, and I worked with this protein TRA-R, which is the homologous of LuxR, particularly focusing on the dimerization interface. So this protein dimerizes, and I studied basically this dimer interface, studying which amino acids were important for dimerization. But why is this protein important? Because when it is activated, it makes agrobacterium tumefacients more virulent. What does agrobacterium tumefacients do? It basically causes tumors in plants, in several kinds of plants. So you can see here a massive tumor. The plant doesn't die, but it has this appendage, which is kind of ugly and all, but it's there. So how does this happen? So it turns out that the tumor is caused by a piece of DNA that is present in a plasmid. So this is, when this was discovered, it was, of course, Nature and Science papers all over the place, because usually you have proteins that cause problems, not a piece of DNA. A piece of DNA of the plasmid inside the bacteria is transferred to the nucleus of the plant cell, and the plants start producing hormones, and the cells start to grow, and that's why you have the tumor formation. I didn't study that. I studied the plasmid. So this plasmid here, which is called tumor-inducing. Replication of this plasmid is controlled by quorum sensing. So when you have quorum sensing on, the rep genes, rep A, B, and C are activated, and more plasmids are produced. So you can see here from a cell that has one plasmid, when quorum sensing is on, the cell has 8, 10 plasmids. More plasmid, more genes, more virulence. So this is basically a dose-dependent thing. You have more genes, the cell is more virulent. It can cause more damage to the plant. But also, you can see here that TRA-R controls this operon replication and this operon in this other side, which is related to conjugation. Not only the plasmid increases in number, but the plasmid, which is like a selfish element, wants to be transferred to other cells that don't have that plasmid. It's a way of spreading the plasmid. So the plasmid is conjugated to cells that don't have this plasmid. So this is what I spent most of my time in my PhD studying these things. Well, quorum sensing controls that. It controls biofilm formation, as you can see here. Biofilm formation is a complex phenomenon. It happens with attachment of cells, growth, formation of microcolonies. When you have these microcolonies embedded in some exopolysaccharide matrix, you can imagine that quorum sensing is going to happen, because all the cells are together, they are close together in some matrix, and they will produce molecules. So the maturation of the biofilm is controlled by quorum sensing, and also the dispersion. So after some time, quorum sensing happens backwards. So it tells the cells to spread. Okay, this place is already well colonized. Let's go, let's break up and colonize new places. So quorum sensing is also involved in this. And how about food microbiology? Because I could be talking about medical stuff here all morning, but the focus of the course is food microbiology. In food microbiology, there are several spoilage phenotypes, or basically enzyme production, that are controlled by quorum sensing. That's very true for Erwinia carotivora. Now it's called Pactobacterium carotivora. Aeromonas, Hafnia, Serratia. But you also have virulence production in several organisms, including Staph aureus, Listeria, Salmonella, biofilm formation, of course, and maybe to compete with one another, because as we're going to see at the end of the talk, we also have this competition between organisms, and besides producing bacteriocins, producing acids, gram-positives can influence the communication of gram-negatives. When they are together, gram-positives basically mutate, or not mutate, but they mute. they silence the gram-negatives because they can break down the signals. So this was a discovery made in the beginning of the 2000, and it has several implications. People are using these enzymes now to interfere with quorum sensing gram-negatives. During my master's, I had the opportunity to be given a project that I didn't like. And I thought, well, my advisor back then, oh, you can work with this. And I was like, oh, okay. I went home and I was like, I don't want to work with this. And analyzing a plate from a friend in the lab, she was working with Pseudomonas, and the agar had casein. And so after 24 hours, we had this Pseudomonas growing this agar, and they were doing nothing. And then 48 hours or 24 hours later, around the colonies, there was a huge zone of degradation of casein. So it was a halo, right? And I was like, well, what happened between yesterday and today? That yesterday there was nothing, and today there is this huge halo. And I was, a few days back, I had a class on quorum sensing. And I thought, well, maybe this is quorum sensing acting because the bacteria now are in high population. Maybe they produce some signal and all of them started producing enzymes. And then it worked. So that was my hypothesis. So I wrote the project. I told my advisor, that's what I want to do in my master's. And this was one thing. And the other thing was, well, this same friend was isolating several bacteria from raw milk. So it was a big PhD project, and she had like a bank of gram-negative bacteria with, I don't know, 200, 300 isolates. And I wanted to ask, well, can this isolate produce homoserine lactone? This is 2004, 2005. There wasn't much on the literature about this. So back then it was new. So what I did was basically, I got some strains, biosensory strains that can detect homoserine lactone. And I asked these biosensory strains if the isolates from milk produced homoserine lactone. So here we have Chromobacterium violations is a mutant that does not produce homoserine lactone. And when I cross this mutant next to a strain that does not produce homoserine lactone, the bacteria is just cream color, no change. But if my isolate produced homoserine lactone, especially, I think it's less than 10 carbons, not more. Okay, it's less than 10 carbons. The culture turns purple. So this is an indication that my strain produces homoserine lactone. And the same is true for this E. coli, which is a mutant that was created to detect homoserine lactones of 4 to 8 carbons with oxo substitutions. And we can see bioluminescence, so this is an indication. And agrobacterium tumefaciens, when it turned blue, in this case, was also an indication of homoserine lactone. So this was the first time that we showed that gram-negative bacteria from raw milk produced an array of signaling molecule. This was during my master's. It took some time to publish. Back then, it wasn't like you do some work today and next month you publish. It took like a year or two to publish. And then follow-up works also showed that this bacteria produced an array of signaling molecules. So a few other students in the lab characterized which molecules were produced and what they were doing. So for instance, Aeromonas hydrophila was using quorum sensing to produce hydrolytic enzymes, basically proteolytic enzymes. So this strain is highly proteolytic. If you basically make a mutation in the quorum sensing system, the proteolysis decreases a lot. I wouldn't say it is completely abolished because there are other systems that control, but it's decreased a lot. So these results confirm the production of several kinds of homoserine lactones in auto-induced too. I left the lab and the quorum sensing work continued in my supervisor lab in Viçosa, Cristina Vanetti. And they found out that there was one gene in Salmonella, which was homologous to LuxR. This gene was not called Salmonella R or whatever, it had a weird name, SDIA, was discovered involved in something else, but it was homologous to LuxR. But when you look at in the genome of Salmonella, you can't find the LuxI gene or the gene that produces homocyanin lactone. So people started wondering, well, what does this gene do? Can it detect homocyanin lactone produced by other organisms? And this is what they found. Emiliani and Felipe Almeida, which many of you know, tried several experiments, and basically the conditions that worked was anaerobic conditions, and they would add either a mix of homocyanin lactones or basically this one, C12, which was the best one, and the strain would go from just attaching to the surface to producing a lot of biofilms. So this was quite interesting. This was confirmed through different microscopies, and this was the discovery that they made, the contribution. Well, very important, because you don't want a Salmonella get into your gut, detecting other gram-negatives and producing massive biofilms in your gut. That would be horrible for your health. Knowing that quorum sensing is so important, how can we interfere with it? So many people start wondering how we could interfere with quorum sensing. So we can interfere in several ways. If we remember, we have the production of the signal. You can have quorum sensing inhibitors that will act at the enzyme that produces homocyanin lactone. You can have quorum quenching enzymes, which will basically be present in the environment and will break down the molecules, or you can have other molecules that will interfere with signal receptance at the LuxR level. Back then, in my PhD, working with agrobacterium tumefacients, I came to know that plants use phenolic compounds for many reasons. One of the reasons is to do what they call allelopathy, in which one plant inhibits the next by producing some phenolic compound which is toxic for that plant. But also, they produce in the environment of the root, which they call rhizosphere, they produce these phenolic compounds which have inhibitory effects over nematodes, bacterial pathogens, except agrobacterium tumefacients in rhizobium. Agrobacterium tumefacients in rhizobium swim towards the plant when they detect the signals. So I started wondering, well, if this happens with agrobacterium and rhizobium, can we use phenolic compounds to inhibit quorum sensing in selected bacteria? The sad part is that I thought that, and the rest of the world also thought the same. So it was not very original in the end. So we started working with the effect of phenolic compounds on quorum sensing bacteria. I had several students working on that in the past, and presently, Emilia, Catarina, and Cristina. So before working with the phenolic compounds isolated by themselves, we started working with plant extracts. In Brazil, we have several exotic plants, and we started working with plants from the Atlantic forest. And we made these extracts, and we started testing them. Well, can they inhibit quorum sensing? So we have the biosensory strains, especially that one that produces the purple pigment. It's very easy to work with when it doesn't die. And then when we added the phenolic extracts, you can see that here's the control, a lot of violacein production. With phenolic extract, very little production, even better than the positive control of inhibition, which is furanone, which was discovered back in 2002, inhibiting quorum sensing. So this was quite interesting. We did that with several plants, and the problem with plants or making extracts of plants or fruits is that the extracts are difficult to reproduce. So you have a production this year, next year, you might not have the same phenolic composition. So we started wondering which compounds are actually inhibiting quorum sensing, because these extracts are basically a soup. They have a lot of stuff. So Emilia and Catarina also started doing some molecular docking work based on the literature. And what we did was basically ask the computer to bind these phenolic compounds to the LuxR receptor and see if they would bind well or not. And basically, based on the scores, the ones that bound the best, then we bought it and we tested in the lab. So here's just a piece of data from Emilia. The work is not published yet. And you can see in this assay, Pseudomonas lasb GFP, without the compound, you have a lot of GFP production. And with the, what is this compound? Resveratrol, it's just an example. You have a very strong inhibition of GFP production, indicating that it might be inhibiting quorum sensing. When she took these selected compounds out of 80 something, 4 were really good, and they inhibit raminolipids, they inhibit protease, biofilm formation, several phenotypes. For instance, here you can see the development of biofilm by Pseudomonas without the compound, so it forms this strong biofilm. I'm going to turn off this light. It's very clear that it's producing a robust biofilm, and as we combine these molecules, and you can have an effect of inhibition or completely destruction of the biofilms. So we started with isolated, with extracts, and then we went on to isolated compounds, and we found out in the end that they work better together, like in the extracts. So it's funny to think that way. Just a piece of data here that I obtained with bicalain. You can see that it really inhibits swarming motility at very low concentration. This is 50 micromolar. In the literature, they use 500 micromolar, 1 millimolar. So we went down all the way up to 50. This is also true for piocyanin production, which is a virulence factor. So you can see bicalain, quercetin, also very good at inhibiting these phenotypes. This is the structure. There was one in vivo study I did. This was just like the beginning. We need to continue. And in this fruit fly assay, we basically infect the fruit flies with Pseudomonas, and we feed the fruit flies with phenolics. I think I fed the fruit fly with just too little phenolics. The concentration was too low. But even at very low concentration, you can see that bicalain at 200 micromolar made the flies to live longer in the presence of the Pseudomonas aeruginosa. You can see that without the compound, the flies are infected, right? So they're going to die because of the infection. So they die faster in the absence of the compound than in the presence of the compound. This needs to be confirmed. There was one experiment I did. Didn't have time to repeat it. But it can be, it's a good indication that it might work in vivo, especially if we increase the dose, because the dose that I used, I had to choose one dose, and I chose this 200 micromolar dose, which might be too low considering the system. So which compounds are most promising? Quercetin, a bunch of people are working with it. A bunch of people are showing promising results, including us. Luteolin, I don't know, it increased, it got better. Luteolin also very good, especially we have a paper on that. And bicalain, which was found to be very good as well. I don't know what I did here. Let's see, okay. So what is the inhibitory mechanism? We propose that these phenolic compounds would somehow exchange or entering the active site of the LuxR protein, and then it would make the protein unable to activate transcription. The Basler group at Princeton, they show that it actually might be in another pathway. So the phenolic compound would not interact in the active site, but would actually interact somewhere else in the protein. So they call it this allosteric inhibition. And they have very good reasons, very good results showing that. So it might be true. But it might also be true that it exchanges or it binds to the pockets of the protein. This needs to be further confirmed. So the last part of the talk, and I'm running late, but Thiago, which is the next speaker, he said, oh, you can grab some of my time. So I'll grab some of your time, Thiago. I'm sorry. But this is going to last 5 minutes, I think. So the next thing that we wanted to do was to study the fermented whey that it's used for artisanal cheese production. The fermented whey in the Canastra region is called pingo. So I'm going to be talking pingo. Basically, it's the whey that is drained from the cheese. It ferments. It has lots of bacteria and phages and everything, but mostly lactic acid bacteria. And the producers use this in the backing slopping method. So they used to inoculate a new batch of milk and make new cheese. And they do this every single day throughout the year. So I looked at the pingo and I thought, well, this is perfect for studying microbial interactions. And Augusto started working with this. And the hypothesis or the title of the project was Population Interactions Mediated by Quorum Sensing in the Endogenous Culture Used in the Production of Canastra Cheese. So what is Canastra cheese? Canastra cheese is produced in Minas Gerais. So here's the map of Brazil in Latin America. Minas Gerais is right there, and Canastra is right here, the Canastra region. We have several regions that produce cheese in Minas Gerais, but Canastra, which is one of the most famous, maybe the most famous one, is right there. So how this cheese is made? Yesterday you had a talk about cheese, but basically, you have milking, filtration, addition of pingo, which is the fermented whey, rennet, coagulation, stirring, whey drainage, and molding, manual pressing. So they do that using cloths. And some of them are having problems in, I don't know how to say that in English, because it's laborious. So they're having problems in their wrist because of that. First salting, flipping, and second salting. And then this is like the nights. So they collect the pingo throughout the night and they use the pingo next day. And the cheese is ripened for only 14 days. It's very low period of ripening. So here is how they collect the pingo in this apparatus. And they use this for production, right? So our hypothesis was that pingo provides a conducive environment for microbial interactions mediated by quorum sensing and quorum quenching as well. So what Augusto did first was to collect a bunch of pingos in the Canastra region, make extracts, and see if there was homocysteine lactone present. And this was the frustrating part of the masters, of his masters, because everything was negative. Until one producer said, hey, I have this pingo here and my cheese went bad. It didn't work. The cheese was terrible. Can you analyze this pingo? So Augusto took the pingo and sure enough, it had homocysteine lactone. So it seems like the presence of homocysteine lactone in the pingo is an indication of bad quality. So unsuitable for production. What Augusto did next was to isolate bacteria from all these pingos and see if these bacteria were able to produce homocysteine lactones. And surely enough, they were. So out of 150 isolates, 39 produced homocysteine lactones. First report of these strains or species producing homocysteine lactone. We need to publish this. And then next we ask, well, okay, there is homocysteine lactone, there is potential for production, but the majority of the pingos don't have homocysteine lactone. What is happening? So the hypothesis was that, well, gram-positive bacteria are inhibiting the production of these signal molecules in gram-negatives. So how do we prove that? So we created a very simple experiment, but at the same time elegant, in which we come, in the first day, we make a circle of the strain we are trying to analyze, the gram-positive strain. In 24 hours, that circle produces, the bacteria, right? Then next day, we come and we make an internal circle of the biosensory strain. So right here, you have the gram-positive. Next day, you have the biosensor, which is gram-negative, and will detect homocysteine lactone in the medium. I should say, right, that we put homocysteine lactone in the medium. So the gram-negative would turn purple because it has homocysteine lactone in the medium, unless the gram-positive breaks it down or interferes in some way. So 4 things can happen. 1, the gram-positive can kill the gram-negative. This can happen, and it did. Another thing that can happen is nothing, right? The gram-positive grows, the gram-negative grows and becomes purple. Nothing happened at quorum sensing level, no interference. Or you can have interference, total interference in the quorum sensing, so the biosensor strain does not produce any pigments. Or you have partial inhibition, which the gram-negative only becomes purple in the center and not at the edge of the colony. So Augusto observed all of that. So you can see here, completely killing of the biosensor strain, no change, the biosensor strain completely purple, completely inhibition of quorum sensing, the biosensor strain, so it's cream, and partial inhibition, where you have only inhibition at the edge of the colony. It's a master, so he didn't have time to study everything. So he focused on these strains. Why? Because we wanted to find out enzymes that inhibit quorum sensing. And because enzymes are big, we hypothesized that in the agar, they wouldn't travel far enough. They would be only in the edge. So he studied several of these isolates. Well, just a summary of these gram-positive isolates. So you have maybe almost 40 isolates that had no interaction, and you had 140 that inhibit quorum sensing in some way, and you had here maybe 30 that killed the biosensory strains. And from these ones, we focused on the ones that had partial inhibition. After a series of experiments that I don't have time to go through, he found out that 28 gram-positive isolates belonging to 4 genera and 8 species produced enzymes that break down the signaling molecules. So all of these species here, first report of lactonase production by Staph Chilosus and acylases by Orius and these other guys. This has yet to be continued. So it's a work that has a lot of potential, especially when we think about this completely inhibition of quorum sensing. Which molecules are being produced that are completely inhibiting quorum sensing in these strains? The strains are there in the lab, and somebody can do a PhD on that. The number of gram-positive isolates without the ability to inhibit quorum sensing gram-negative bacteria. Why this set of experiments? Basically, to show you that, you remember that sample that was bad for cheese production? We call here Pingo 11. When Augusto analyzed the gram-positive strains isolated from there, 95% of those isolates did not inhibit quorum sensing at all, indicating that maybe inhibition of quorum sensing is important for cheese quality. It could just be by chance, but that's a good, indication that it is important. So this work reviewed a highly competitive ecosystem through the exchange and interception of chemical signaling in the Pingo. Is the first study to explore microbial interactions mediated by quorum sensing the microbiota of artisanal cheeses, and possibilities for various studies. Potential applications. We envision applications in packaging. There are some studies that we are doing on that. I don't know where Katarina is. She should be here, and she's going to be doing that. Detergents and sanitizers. Prevention and treatment of foodborne diseases, maybe. Can we eat more foods that have more phenolics? Can that be good for our health? We know that it can, but could it possibly prevent foodborne diseases on quorum sensing inhibition? Well, we don't know yet. Many possibilities in the medical field, especially when you think about catheters. You could attach quorum sensing enzymes, you could attach quorum sensing inhibitors in these catheters and avoid colonization by Pseudomonas aeruginosa, for instance. So there's a lot going on. There's a lot of patents on this as well. People working in the US, they have patented a lot of molecules and applications. So this is our team. This work was done by students, not by me. So most of it, at least a few figures was by me, but most of the time it's the students that do. And I would like to thank my partners and my funders and you for your attention. This is it. We don't have time for a break, right? We don't have questions in the chat. Maybe we can entertain questions here, one or two. Yes. Do we have microphone for him? But the people in the audience can't, so please.
Okay. Okay, thank you, Professor, for the wonderful presentation. Well, my question here is, how does current sensor differ between gram-positive and gram-negative bacteria?
Yeah, I didn't have time to go through that, but gram-negatives communicate through acylhomocysteine lactones. Gram-positives communicate through autoinducer peptides. So small peptides, they have a gene coding for them. Some bacteriocins are considered colon sensing signals as well. Not all of them, but some are. And you have other molecules as well, but these are the two most important groups. Okay. What is that? Not lactones, the acylhomocysteine lactones. I don't know why they're important. That's a good question. I mean, that's why do you have, why are they the signaling molecules and not other, right? I guess that could be an answer, a question. As I said in the beginning, the substrates are very common in the cell. So you have, you know, ácido gráfico, fatty acids, and you have one amino acid. So what basically the enzyme does is to make a ring out of the homoserine, of the serine amino acid, and attach a different fatty acid to it. So agrobacterium does that with a molecule with 6 carbons. Pseudomonas uses 2 with 4 and 12. Chromobacterium with 6. So you have different kinds of lengths of these signals, which make the signal specific for that particular species. So yeah, you have variations in the signals that make them specific, and this could help the organism. But why the choice for that? It's a question that doesn't have an answer. All right, thank you guys. Now we go on with the next speaker. I don't know if he's online already. Thiago, are you there? Hello, Thiago. Can you hear me?
Yes, I can hear you.
Hi. Good morning.
Morning.
It's a pleasure to welcome Thiago Santos to our course. I'm going to just introduce Thiago a little bit. He's going to be talking about the envelope, please. Strategies to selectively manipulate the microbiome. Thiago is at the Northeastern University in Boston, United States. I could just read this, or I could say something more personal. I know Thiago for a long time. He's a very good friend of mine. We met at the University of Viçosa, and then at Cornell when he spent some time there, and I was doing my PhD. Then my visiting, as a visiting professor at Harvard, Thiago was a postdoc there, so we have a bunch of history together. So Thiago is graduated in biological sciences from the Federal University of Viçosa, where he also pursued a master's degree in microbiology. He then went on to a post-bachelor's degree at Cornell University, where he stayed for a few years, and he went on to pursue a PhD in microbiology at the University of Wisconsin in Madison. And then he got a postdoc in the area of protein biochemistry at Harvard University at the Department of Chemistry and Chemical Biology. He's a Howard Hughes Medical Institute scholar, which is a very big deal. And as of January 2024, so just this year, he's an assistant professor in the Department of Chemistry and Chemical Biology in the Department of Biology at Northeastern University, Boston. So, Thiago, welcome, and the floor is yours.
Thank you so much. Thank you, Uelinton, for the introduction. Can you see my screen, like the full screen?
We can. The volume is just a little too low. I'll ask the technical team to increase the volume, please. You can speak, Thiago.
Can you hear me better now?
Can you hear him better? Yes, people are saying yes. Okay.
Okay, good. So thank you so much, Uelinton, for the introduction. And like you said, I did study microbial interactions for several years during my training, but more recently I switched a little bit to biochemistry. So I have some bad news to deliver for those who are really looking forward to microbial interactions talk. I'm going to talk way more about biochemistry today, but maybe at the end, I'm going to give you one slide where you can see how my work fits into microbial interactions, which is the topic of this of the lectures today. But what I'm going to show you today, like Uelinton said, I just started my lab, about 6 months ago, almost 7 months now. So most of the things that I'm going to show you today is work that I did during my postdoc, and in which I studied how bacteria make this essential piece of their cell envelope, which we call the outer membrane. I'm sure a lot of you in the audience are microbiologists and probably familiar with these terms. But if you're not, don't worry. I'm going to give a quick overview on everything that you need to know. So specifically, when it comes to the outer membrane, I'm going to discuss how bacteria make proteins, transmembrane proteins in this outermost layer of this organelle. And we are interested in this problem because my lab is still studying this problem, and we are interested in this problem because these proteins are essential for survival of all gram-negative bacteria. And we think that it is an important target for the development of new antibiotics. So for most of my talk, I'm going to show you how we think that the chemistry that is involved in folding proteins in the outer membrane of bacteria occurs and how we get information about this process. Okay, so, and like I said, toward the end, I'm going to talk about, how my lab is now trying to use bacteriocins to target this process, outer membrane biogenesis, in gram-negative bacteria in a very selective way, which bacteriocins allows us to do. So, this is probably not a news to anyone in the audience, but I must emphasize that, there is a big crisis, related to the rise and spread of antibiotic resistance among pathogenic bacteria, which is a major public health crisis. And this graph that I'm showing you here, it's a study that was published about 2 years ago now, and in which they estimate that about 5 million people died of a drug-resistant bacterial infection in the year 2019. And these organisms that I'm highlighting here, the gram-negative bacteria, which are the ones that I'm highlighting in this graph now, are the most problematic ones, simply because we don't have antibiotics that are effective at killing these organisms. And one of the reasons that it's so hard to treat infections caused by gram-negative bacteria can be partially explained by the architecture of the cell envelope of these organisms. So most bacteria can be separated into these two major types based on the architecture of their cell envelope. So we have here, can you see my my pointer? I can't hear, but I'm going to assume you are seeing the pointer. On this side, I have gram-positive cell, in which you have an inner membrane and a thick layer of peptidoglycan. And the cell envelope of gram-negative bacteria also contains the inner membrane, a thin layer of peptidoglycan, but in addition, it has this second membrane that we call the outer membrane, highlighted in green. And this outer membrane is exactly why it's so much more difficult to treat infections caused by gram-negative bacteria. This outer membrane is highly impermeable to antibiotics. And I can show you that it is impermeable just by doing a very simple experiment. So here I took a gram-positive organism, Staphylococcus aureus, and a gram-negative organism, E. coli. And you can see that in the absence of an antibiotic such as vancomycin, both cells grow fine, but in the presence of vancomycin, only E. coli is able to grow. Staphylococcus aureus is completely inhibited in this case. But one thing that I should probably tell you at this point is that vancomycin, which is a very important antibiotic, targets the biosynthesis of peptidoglycan, which both cells have. So the reason why E. coli can grow in the presence of vancomycin is because vancomycin and many other clinical antibiotics cannot penetrate this outer membrane to reach the target. And if we zoom in here in the outer membrane, this is what we're going to see. Unlike the inner membrane, which is contains phospholipids in both the outer leaflet and the inner leaflet, the outer membrane is this asymmetric lipid bilayer. It contains phospholipids in the inner leaflet, but it contains this looking lipid called lipopolysaccharide in the outer leaflet. And this asymmetric distribution of lipids is a key to create this impermeable barrier to antibiotics. In addition to the lipids, the outer membrane also contains different types of proteins. It contains these proteins that we call lipoproteins, that you can see here. And also it contains these transmembrane proteins, these large transmembrane proteins that we call beta-barrel proteins. And even small changes in the composition of this outer membrane can be detrimental to the cell. So, as you can probably imagine, it's not easy to build this organelle, this outer membrane. And one challenge to making this outer membrane is that all its components, both the lipids and the proteins, are made in the cytoplasm of the cell, and they need to be transported all the way to the outer membrane. And this is not an easy task for the cell. But remember, like I said, this is a very, very essential organelle, this outer membrane. So because bacteria, gram-negative bacteria, care so much about this outer membrane, they have evolved several machines to build this outer membrane and to make sure that this organelle is properly assembled. So there is this system called the LOL system, which is important for transporting lipoproteins from the outer leaflet of the inner membrane to the inner leaflet of the outer membrane. There is this other system called LPT system, which is important for, which is important for transporting lipopolysaccharides to the outer leaflet of the outer membrane. And finally, there is this other system that we call the BAM complex, which takes unfolded proteins from the periplasmic space, folds these proteins, and puts these proteins, these folded transmembrane proteins, into the outer membrane. And this machine, the BAM complex, was the focus of my research during my postdoc, and now it's the base for one of the branches of the research in my independent lab. So when I was studying this complex, we wanted to understand how this machine takes an unfolded protein from this aqueous environment, folds this protein, and puts this protein in this hydrophobic environment, which is the outer membrane. But why this is important, right? Like, why should we care about this? So there is a very practical reason. If you think about development of antibiotics, this could be a good target for the development of new antibiotics, so you can kill pathogenic gram-negative bacteria. But there's also very fundamental importance to this problem, which is the central component of this machine that I'm showing you here is conserved all the way from bacteria to humans and plants. It's in the outer membrane of mitochondria, chloroplasts, folding beta-barrel proteins in those organelles. And I think everybody here agrees with me when I say that any biology that is conserved to this level must be very important. So there's both practical applications and fundamental applications to this problem. And here is what a beta-barrel protein looks like. As you can see, it's a beta sheet, is a sheet of beta strands actually wrapped into a cylinder, and they're connected by these loops. And if you rotate them, you can see that they, look like just like a cylinder. And the way that the amino acids are organized in this protein is that hydrophobic residues are exposed to this the hydrophobic environment of the membrane, and the hydrophilic ones have their side chains pointing to the lumen that we call of this protein, where there's water. And from a chemical perspective, it's not easy to fold these proteins, right? Because you cannot fold a transmembrane protein outside of a membrane. Otherwise, this protein would be inside out. But you also cannot put an unfolded protein in the membrane because you'd have a lot of unsatisfied hydrogen bonds exposed to this hydrophobic environment of the membrane, and this would also be very bad. So the BAM complex that I just showed you here has to do these two tasks, right? It has to fold these proteins, this unfolded proteins that it receives from the periplasmic space, and then insert these proteins at the same time into the membrane. So it's a very challenging task. Another challenge associated with this process is that these substrates for this BAM complex, they're very different, right? So you can see here that there are like some big proteins, some small beta barrels, some beta barrels that associate with other beta barrels to form these oligomers, and then even some beta barrels that have other lipoproteins living inside of them. So there's a diversity of substrate, and yet there's only one machine to fold all these different substrates. And so you can think that BAM is not your average enzyme, right? Like, how many enzymes can you think of that has more than one or two substrates, right? So the BAM complex has hundreds of different substrates, and it must be able to fold all these different proteins correctly and put them in the outer membrane. And maybe some of you are thinking now, well, if these proteins, these substrates are so different, they must all have different requirements for folding. And I agree with that. However, because there's only one machine to fold all these different substrates, there must be features that are conserved among all these substrates, and these are the features that we were studying. What are the conserved features during the folding of all these different substrates that this machine needs to recognize in order to fold them? And one even more fundamental question to this process is, it's what steps in this process needs to be catalyzed, right? Like when you think about folding a protein, what steps of the folding process requires a catalyst, which this protein is. And this is where my journey starts. So this is what the complex looks like. It's a 5-protein complex. There's this central component called BAM-A, which itself is a beta barrel, as you can see here. And it has 4 lipoproteins: BAM-B, BAM-C, BAM-D, and BAM-E. So one of the challenge to studying this process is that we need to be able to study this enzyme, this catalyst, in the presence of a substrate, in the presence of a beta barrel, to understand how the process happens. And like I showed you, there are hundreds of different substrates. We have to choose one in order to do biochemistry and understand how this process happens. And this is one of the substrates that I chose. The reason why I chose the substrate, this protein I showed you before, this is it's a beta barrel protein, which is part of this trans-envelope complex that is important for transporting LPS, lipopolysaccharide, to the outer membrane. So you can say that we're studying the folding of the protein that makes this outer membrane. And one of the reasons why we chose this protein is there are several reasons. One is because it's a very desirable protein for biochemical studies. If you imagine this beta barrel, if you just open this beta barrel, this is what you would see. LPTD is one of the largest beta barrels in E. coli, for example. It has 26 beta strands, and it's the slowest substrate to fold. So for most beta barrels in E. coli, it takes about a fraction of seconds to fold other beta barrels. But LPTD, which is the substrate that is important for in the transport of LPS in the cell, it takes about 20 minutes. So the machine, that BAM complex, takes about 20 minutes to fold this protein. So it's a very it's a slow substrate to be folded. There are some barriers that need to be overcome to fold this protein. A second feature of this protein that is also desirable for the kinds of studies that I'm going to show you is that the oxidation state of this protein can be used as a proxy for folding. So for example, here you can see that LPTD has 4 cysteines, if you look here. And when this protein is fully folded, they form 2 long-range, non-consecutive disulfide bonds, in which the first cysteine in the protein forms a disulfide bond with the third cysteine, and the second cysteine forms a disulfide bond with the fourth cysteine. During the folding process, there is an intermediate state in which the first cysteine forms a disulfide bond with the second cysteine. And we can see this on a gel. So if you take a sample that contains this protein called LPTD, which is a beta barrel, and it is a substrate for the BAM complex. And if you add a reducing agent like beta-mercaptanol, you're only going to see one band because all the cysteines are going to be reduced. However, if you don't use a reducing agent, you're going to see all these 3 bands, right? The reduced band, the intermediate one in which you have this configuration of cysteines, and the fully folded mature protein in which you have this different configuration of cysteines. And this is very important for some experiments that I'm going to show you later on. So just remember this configuration here. If you have the protein in the non-reducing state, you can see 3 bands on a protein gel. And that tells you whether or not this protein is properly folded and fully folded. Okay, and the third thing is that, the third important feature is that because LPTD forms this transporter that delivers lipopolysaccharides to the cell surface, any problem in the assembly of this protein results in an outer membrane that it's leaky, that is permeable to antibiotics that normally do not penetrate the cell. So if you remember earlier, I showed you that if you take E. coli cells and you plate them in vancomycin, like I'm doing here, they don't really care. They grow in the presence of vancomycin. But if you make defect in this protein, LPTD, some sorts of defects that will prevent this protein from being properly folded, you can see now that cells become highly susceptible to this antibiotic vancomycin. So again, highlighting this notion that if we are able to prevent the folding of the substrate, for example, in other substrates, we might be able to make gram-negative bacteria leaky and permeable to antibiotics that normally do not penetrate the cell. Okay, but although I said that this protein is a slow-folding substrate, that it takes about 20 minutes to fold this protein, this is not enough to do biochemistry. So for those in the audience who do biochemistry, sometimes we need more time than 20 minutes to study a process. So to do that, we had to engineer the substrate and make modifications to make the folding process even longer than 20 minutes. And this is one of these mutants that I made. As you can see here, so this is the whole protein. Well, it's like part of the beta barrel domain of this protein called LPTD. And the only modification that I did was to delete this residue here, this aspartate, at position 330. So when I do this, when I remove that one amino acid, so think about this, this protein has about 800 amino acids. Just by removing one amino acid, I'm able to slow down even more the folding of this protein. And I can see, and I know that this protein is stalling in BAM during its folding because we can see accumulation of the protein in the machine. So if we use, for example, this cross-linker called DSP that I'm showing you here, and we treat cells with this cross-linker, we can pull down this complex in blue, the BAM complex, and we can see whether or not the protein is stalled in this complex. So here is, for example, it's one example of an experiment. If we have a wild-type protein being folded by the BAM complex, and we pull down the BAM, in this case, BAMA, one of the components of the complex, you don't see the protein LPTD stuck in the complex. However, if you make that one mutation in which you delete one amino acid, that residue, the aspartate 330, now you can see this band here indicating that this mutant protein, this mutant version of this protein, is stuck inside the BAM complex. It spends enough time in the complex that you can capture these interactions. So that tells us that the folding of this protein has been slowed down by the deletion of that one amino acid. And we can also show that because we're slowing down the folding of this substrate, now these cells become very sensitive to vancomycin. So if you remember, wild-type cells, they grow well in the presence of vancomycin, but if you mutate that residue, now the cells become very sensitive to vancomycin. So that's pretty cool. And we can also do pulse chase experiments to compare the actual rate of folding to show that this mutant, this LPTD mutant that folds slower than the wild type, is actually folding slower. It's not some other problem that is happening. So for these kinds of experiments, for those, I'm sure Uelinton has done several of these. You have, proteins being made in the cell, and one thing that you can do is we pulse the cells, we treat them with radioactive, in this case, amino acid methionine, and the cells are going to start making radioactive proteins. And then next, what you do is you chase this culture by adding unlabeled methionine. So all you're doing here, now that you have an excess of this unlabeled methionine, the cells start making again proteins that are not radioactive. So what you did here is you created a population of proteins that are radioactive, and you can now track the folding of this group of proteins over time. So we did that, and if you remember that experiment that I showed you a few slides back, when in this case here, we collected samples every 5 or 10 minutes. And you can see that it takes about 20 minutes to see the same amount of partially folded protein and folded protein. So if you remember that piece of data that I showed you before, we can track the folding of this protein just by looking at those bands in the gel. So here at 20 minutes, you can see that you have about the same amount of partially folded and folded protein. But when we delete that one amino acid in the same protein, this very important essential protein called LPTD, now it takes about 2.5 times longer to see this conversion from the partially folded to the fully folded protein. So here we're showing that deletion of that one amino acid changes the rate of folding of this mutant substrate. So the next thing that we decided to do was to look at the folding pathway. We asked, what's the folding pathway when the substrate is being folded by this complex? In other words, what parts of the substrate and the machine contact each other during the folding process? So to do this, we use this other strategy in which we replace specific residues in the protein with this unnatural amino acid called PBPA, which is short for para-benzoyl alphenylalanine. And this is an unnatural amino acid. We don't have proteins that have this amino acid, but we use an engineered system in which we're able to incorporate this amino acid in one specific position of interest in your protein of interest. And what happens is when this protein is being folded, if you you expose your culture to UV, what happens is that this PBPA will form a reactive species, and this reactive species will cross-link to molecules that are about 4 angstrom from the polypeptide backbone. So in this case, LPTD in this case would form one cross-link to BAM if this distance between this unnatural amino acid and the BAM complex is less than 4 angstrom. And if we pull down BAM, we can see whether or not this protein comes with it. And here's just one example of how this experiment would work. So here I have the wild type LPTD. I put that unnatural amino acid at position 468 in BAM-A. And when the protein is trying to fold a wild type substrate, even in the presence of UV, you don't see anything. And that indicates that the protein is just being folded quick, that you don't quick enough that you won't be able to capture any interaction. However, when you delete this amino acid in that substrate that is slow to fold, now you can see the interaction. And that means that when the substrate is being folded, this protein comes very close to this residue position 468 of BAM-A. So imagine if you do this for hundreds or of residues, both in the protein and the substrate. This was the picture that we're able to build doing these kinds of experiments. And what we saw, this basically, I'm just summarizing several months of work and hundreds of in vivo photo cross-linking in one simple cartoon. And what we learned here is that the C-terminal region of the substrate formed this continuous beta sheet. So here we're looking from the top, right? BAM-A is here, and the substrate. There is the formation of this continuous beta sheet between the substrate and the machine. And we see that there's a large portion of the protein. There's this extensive region of the substrate inside, in the interior wall of BAM-A, as you can see here. And this kind of gives an idea, a suggestion that maybe folding of substrates is happening inside this complex, this BAM-A complex. And an important question that emerged from these studies was if the interior wall of the BAM-A serves as an active site for to catalyze folding of the substrate. So to address that question, what we did was we designed a genetic screen, and we found one suppressor mutation, one mutation in the interior wall of the BAM machine that was able to accelerate folding of that slow folding substrate and rescue folding of that slow folding substrate. So if you remember, you've seen this before. If you grow E. coli cells, wild type E. coli cells in the presence of vancomycin, they don't care. Vancomycin cannot penetrate this outer membrane. If you slow down the folding of one of these beta barrels by deleting that one amino acid, you now make cells very susceptible. But if you now mutate the machine, and this is why we call it compensatory suppressor mutation, in the wild type, you don't see any difference, but now that suppressor in the machine is able again to fold the mutant substrate, and the cells, even with the mutation, are resistant to vancomycin. So if you look at the this pulse chase experiment that I showed you, it takes about 20 minutes to see the conversion here, 2.5 times longer in the mutant substrate. When we use the suppressor machine, the machine that is now able to fold that slow folding substrate more efficiently, you see that now it takes about 25 minutes for us to see this 50-50% conversion. So this experiment shows that we can restore the rate of folding of a slow folding substrate almost to wild type levels. And this argument supports this hypothesis that this interior region of the machine where we found the suppressor, I'm going to go back here just to highlight it again. So in the interior wall of the machine where we found the suppressor is important for catalysis, for accelerating this process. And this is where we stopped. We are now trying to understand, we have some work that is about to be published, hopefully soon, to understand what exactly this interior region of the barrel, how this interior region of the barrel is accelerating folding of proteins, of beta barrels in general. So just to briefly summarize what I've shown you so far is, we were interested in understanding how these beta barrel membrane proteins are folded. And, they're very important for maintaining this impermeability of the outer membrane of gram-negative to antibiotics. We found that the assembly of substrate is catalyzed in the interior wall. So that work has been published. And now something that we are working and about to publish soon is that we have some evidence that I'm not going to go over here to support this notion that this region here is very important for folding, for forming a rudimentary beta sheet that will initiate folding of substrates. And I'm happy to discuss more details about these experiments in the Q&A at the end. But, we have to talk about interactions and how does my work connect with microbial interactions. So I hope that by now I've convinced you that this is a problem, right? The outer membrane of gram-negative bacteria is highly impermeable to antibiotics. And what if we're able to make this outer membrane leaky now? So one candidate that we are exploring now in my lab is the use of bacteriocins. And how can we use bacteriocins to design inhibitors that can make this outer membrane leaky? So our premise is that if we make this, if we find inhibitors that target these machines that fold beta barrel in the outer membrane of gram-negative, that in itself will be an inhibitor, but it can also synergize well with other antibiotics that we already have, but are ineffective against gram-negative bacteria just because they can't penetrate this outer membrane. So yesterday, several people talked about bacteriocins. Wellington mentioned today bacteriocins, but I just want to remind everyone is this vast collection of peptides and proteins produced by bacteria, sometimes other organisms as well, and selectively kill bacteria that are closely related to the producer strain. This is the classic definition, right? And unlike small molecule antibiotics, many bacteriocins are narrow spectrum. Not all of them, but many of them are. And now that we know the drawbacks of small molecule, broad spectrum agents on the microbiome and the spread of antibiotic resistance, that's one of the reasons why we're so motivated to study bacteriocins for the development of what we like to call precision drugs, drugs that can target specific bacteria in a complex community, such as our microbiome. So we're performing biochemical and structural studies that will teach us how gram-negative bacteria assemble these beta barrier proteins in the outer membrane, or how they assemble their outer membrane, and how we can use bacteriocins to make narrow spectrum inhibitors of this process. And if you think about the relevance of these studies, not only for treating infectious disease, but if you think about bacteria that are related to carcinogenesis, this has far more applications than simply controlling infection, treating infectious diseases. So here, just to show that, again, unlike antibiotics, bacteriocins are able to differentiate sometimes between different strains of the same species. And I think this is fascinating when you think that bacteriocins, just like small molecule antibiotics, target all these essential processes that are conserved in all cells. So one question that keeps me up at night sometimes is, how come the bacteriocin targets these exact conserved complex processes, but they're able to differentiate between different organisms? What kind of chemistry are they targeting that is not conserved in this process that are highly conserved? So I don't know if that makes sense to anyone, but the idea is that we want to understand how these bacteriocins work, and what chemistry of outer membrane biogenesis they are inhibiting. And there's precedent for that. Also, like I said, this is our premise, right? If we can inhibit this complex from functioning properly, we can make this outer membrane leaky to other things that normally don't penetrate the cell. And if you think about efflux, right, several bacteria able to pump drugs out of the cell, you're going to be overwhelming cells with the influx of antibiotics. And even if you're able to pump drugs out of the cell, that's going to be hard for them to survive some strategy like this. There are precedents for that. So, so this is work that was published about 5 years ago now, by actually one of my colleagues here in Northeastern, Professor Kim Lewis and his group. They discovered this compound that they named deribactin, which is a heptapeptide, shown here, and produced by this bacterium called Photorhabdus, which is a member of the microbiome of enteropathogenic nematodes. And you can see here that they are bactericidal, and they cause these interesting phenotypes when you treat cells. Like the surface of the cell seems like very rugged, and they published a few years later, now it's been 3 years, that actually this compound, deribactin, actually inhibits this protein that my lab is very interested in studying, BMA, and it does that by mimicking a beta strand. So in this case, it tricks this protein into thinking that it's going to fold a substrate, and it doesn't, right? It's not a substrate for BAM. And what it does, it mimics a beta strand, and it forms this rigid beta strand that interacts with BAM and prevents BAMA from receiving substrates and from folding beta barriers. And there are some other interesting examples too. So these are this is one of the examples that my lab is interested in studying for Pseudomonas. It's a highly selective protein. In this case, it's not a peptide, it's a protein that is produced by Pseudomonas, and it targets other Pseudomonas. So here is just one experiment from this work that was done by the group of René de Mott in Belgium. And here, just to show you, a strain of Pseudomonas, PA01, is very susceptible to this lectin-like bacteriocin PIO-L1, as you can see here, but not to PIO-L2. If you delete this gene, BAMA, from these cells, and now you overexpress this in trans, here you're overexpressing the same BAMA from another, from Pseudomonas PA1, so it's basically expressing the native gene. You can see that the cells are susceptible. if you expressed an empty vector with that BMA being providing trans, the cells are still susceptible to PIOL1, not to PIOL2. However, when you express BMA from another strain of Pseudomonas BR670, which is susceptible to POL2, now this strain that produces two different types of BAM is susceptible to two different types of lectin-like bacteriocins. And if you just express BR670 BAM-A in PA01 background, you completely change the susceptibility profile of that bacteria. So I think this is a great experiment to show that how these bacteriocins are specific, right? So these are all Pseudomonas, but you just express in different targets now, and they're able to differentiate between two different BAM-As, which are highly conserved in all gram-negative. This is the other example of bacteriocins that we are studying, and I'm gonna tell you later why we're interested in them. But these are called bacteroidectocins, or BD for short. It's a family of small peptides between 42 to 53 residues, and they're produced by and target members of the phylum Bacteroidetes. And so here's just one example: Parabacteroides johnsoni, wild type strains that are susceptible to BDA. You can get mutants, and they happen to be in BAM-A, surprise, surprise. And now these mutants are no longer susceptible, and these strains are not susceptible to BDD, which is just a variation of these bacteriocins. And Bacteroides fragilis, sort of resistant to both of them, but if you mutate BAM, which they claim is and they show genetically that it is a potential target for these small peptides, the cells become resistant. And here they did this cool microscopy experiment in which they treated cells with a probe that was inspired on these bacteriocins, and they saw that these probes accumulated in the membrane, and they can see that when they use this dye FM464, which is a membrane stain. And when they merge, they can see that they merge perfectly, indicating that this membrane is somehow making its way into this probe inspired on bacteroidectocins is making its way to the membrane. And here's electron microscopy. You can see these defects in the outer membrane of the cell, as opposed to untreated cells that have not been treated with the bacteriocin. So there are a lot of evidence, a lot of genetic evidence. So these two studies provide a lot of genetic evidence that BAM-A is the target of this protein. However, there's no biochemical evidence. There's no studies showing exactly how these small peptides and proteins interact or bind, inhibit BAM complex, and this is one of the branches of research in my lab. So we're trying to define now what features of the protein, the complex, the BAM complex, are targeted by these bacteriocins, and what features of these bacteriocins are important for targeting this machine. And like I said, here in this case, we're using two complete unrelated organisms, Pseudomonas and Bacteroides. One is like a soil organism. It's a very important pathogen. It's one of the priority pathogens according to the WHO. And Bacteroides, which is one of the most prevalent organisms in our gut microbiome. So if you think about what we can do with this bacteriocin, is just, the options are endless, right? We can use them to design peptides that will inhibit this complex to treat infectious diseases, but we can also use them in this case, for this particular case here, to as tools to manipulate the microbiome, right? To target specific strains in our microbiome. And for example, avoid dysbiosis. I think this is my very last slide. I show you like one example that we can, how this could be used. Another thing that the lab is doing, we're trying to develop a molecular level understanding of how these bacteriocins inhibit. Like I said, one of the things that we're doing is trying to get structural information of this complex bound to these proteins. This is going to be important to help us understand the spectrum of inhibition of these bacteriocins, how organisms acquire resistance, how we can manipulate the spectrum to make these things, more broad spectrum or even narrower spectrum than they already are. And we can also use these studies to design probes, in this case, fluorescence probes, inspired by these bacteriocins, and design assays, for example, fluorescence displacement assays in which you have probes that bind to the complex, and we can do high throughput screen to find small molecules, drug-like molecules that will inhibit, that would displace, first of all, this probe, and are likely to inhibit this complex in a similar fashion as these bacteriocins. I think this is probably my last slide, but like I said again, so this notion of using these bacteriocins is highly selective proteins and peptides that target an essential component of the cell envelope of gram-negative bacteria. We could use that to, in this case here, you have a healthy gut. And when there is dysbiosis, pathogens can expand. But if we're able to design bacteriocins that are very selective for some type of pathogens, we can restore the healthy gut without disrupting essential important interactions in the microbiome. And then beyond BAM, another thing that our lab is doing right now, actually with a student who is visiting our lab from Brazil, we're trying to identify and characterize other bacteriocins for microbiome. So things are unknown that, we're just doing a bunch of genome mining and finding these interesting small peptides that not only act as antimicrobial compounds, but also have impact on cell-cell communication, like Uelinton was saying, current sensing, defense against bacteriophages, which is another big topic of research, and horizontal gene transfer. I think I'm at time, exactly. I just want to thank all these fantastic group of scientists who have been working with me. This is my lab right now, my current lab at Northeastern. This is our lab right here on the 5th floor, one of these two. And my group, where I did my postdoc, and the folks that help with the imaging at the Cryo-EM Center. And if we have time, I don't know if we have enough time, but I'm happy to answer any questions that you might have. Thank you very much.
Can you hear me?
Yes.
That's a very fancy building. I want to visit it.
Yeah, you should come.
Congratulations, Thiago, for your talk and your work.
Thank you.
I mean, it's impressive, especially working with membrane proteins. It's very challenging. So I have some experience with biochemistry. With cytoplasmic protein is already challenging. I can't imagine with from the membrane. We have some questions from the audience. I also have a question that I'm trying to put together the mechanism. So vancomycin enters through.
Yeah, so I understand.
Mutation. I didn't get that part.
Right. So we actually still don't understand how vancomycin enters the cell. This is another big question, how antibiotics enter the cell, because that mutant LPTD, delta D330 that I showed, is still able to transport LPS. So those cells still have LPS. So technically, the lipid bilayer is still impermeable. So we think that what we think happens is when that protein gets stalled inside BAMA, that's where antibiotics are able to go through. So we think that only by slowing down the folding of substrates, we can create this opportunity for antibiotics to penetrate. But that's a great question. We still don't know exactly the answer. This is just where our working hypothesis.
It makes sense. Makes sense now. Thanks. So a question from Slav. Most of the examples that you have showing are about bacteriocins from gram-negative versus gram-negative targets. Mm-hmm. Do you have any experience about bacteriocins from gram-positive versus gram-negative?
No, but that's a very interesting question. Yeah, I've never seen. No, so these three examples that I showed, you're right, are all gram negative. Never thought about that. Maybe, I don't know, with that, like I said, one of the things that we're doing, like this, mining metagenomes, maybe we'll find something. But no, that's a very good point. Yeah, very good point. Thank you.
There's one more question. I think it's the last one because we're running a little late. What is your opinion on potential resistance to bacteriocins in the near future?
I think that's the $1 million question, right? I think bacteria, it's this endless battle between finding a new drug and then getting resistant, not down the road. But I think bacteriocins give this opportunity to compared to small molecule antibiotics, limit the spread of resistance because, again, you're not exposing, like broad spectrum antibiotics, right? You're not killing everything and giving other organisms opportunity to develop resistance, to mutate and develop resistance, and you spread that resistance horizontally. So I think the idea of developing inhibitors that are, what I call precision inhibitors that will target specific bacteria will lower that chance. But I think eventually microbes will find their way and develop resistance. And that's what, that we're going to keep looking for other inhibitors.
Yeah, it's a never ending battle.
Yeah.
Well,
although I have to say there are some, one of my colleagues here, they found this compound called teixobactin, which they haven't found resistant yet. And everybody's kind of, on their toes, like, just waiting for.
Yes, just wait and it's going to appear.
Just waiting.
Well, let's thank again, Thiago, for this brilliant presentation.
Thank you so much, everyone. Thank you for the invitation.
Thanks a lot. I think we should take 3 minutes break at least. And we're going to start with the next presentation. There is goiabada on the side if you guys want some sugar to activate your brains. And we'll be back at 11:05 Brazilian time. All right. Welcome back. Alessandra, can you hear me?
Yes.
And we can hear you. Great.
Yeah, it's good.
Okay, everyone. I have the pleasure to introduce you to Alessandra Mello. Alessandra is at Laval University. She's going to be talking to us about phage-bacteria interactions in fermented foods. As with Thiago, I'm also good friends with Alessandra, so I can read her bio here, but also I would like to share that I know Alessandra for a long time, and if I'm not wrong, I think I taught chemistry at the pre-vestibular thing.
Yeah. Yes.
Back in the day. I don't remember which year it was, but she was preparing for the vestibular, which every Brazilian knows. And there was this program that I thought, and it's great to see Alessandra flying away very far now, all the way in Canada. So Alessandra has a BS in home economics from the Federal University of Viçosa. You can see that it's common among many of us here, coming from Viçosa, where she started her interest in microbiology. She then went on to a master's degree in agricultural microbiology at the Federal University of Lavras, which is also in Minas Gerais. And then she pursued her PhD in microbiology at the University of Laval, where she studied biology, genomics, and diversity of Brevibacterium phages. And she's currently now a postdoc at the University of Laval at the Department of Biochemistry, Microbiology, and Bioinformatics in the area of genome engineering of phages. So we're going to hear about a lot about phage and bacteria interactions. So Alessandra, once again, thank you so much for clearing your agenda and having a time with us. I hope you're enjoying as well the talks, and we're going to enjoy your talk now. So the floor is yours. Thank you so much.
Thank you very much, Wellington. Thank you very much for all the organizing committee for inviting me for this presentation. So I will put my slides in presenting mode. So can you see it in presentation mode?
Yes, we can.
Yes. Okay.
My audio was off, but yes, we can.
Okay. So put it again. Oopsie. I'm sorry. Yeah. Okay, that's it. Yeah. So as Wellington said, I'll be presenting about phage-bacteria interactions in fermented food, and it's a great pleasure for me to be here today to present some phage work for you. So the outline of my presentation, I will start with a brief introduction, and then I will talk a little bit about bacteriophages, and then I will follow with phage-host interactions, in which I will give you some examples of interactions of phages with Lactococcus lactis, Streptococcus thermophilus, and Brevibacterium rantiacum. And I will finish with the take-home message. So if you were here yesterday, you know a lot already about fermented foods. You know that we can transform raw materials through the metabolic activity of microorganisms. We have several different products that can be transformed. So you have several fermented foods that can be generated from these products. So in dairy, you have cheese, you have yogurt, you have kefir. In vegetables, you have kimchi, pickles as examples. You have in beverage several different beverages like kombucha, beer, wine. And you have several microorganisms that are important in the context of food fermentation. But one group that is very important is lactic acid bacteria, or LAB. And these are key players in the fermentation process as they convert sugars into lactic acid. And they are responsible for enhancing the flavor of the food and also to contribute to the food preservation. And some examples of LAB bacteria is Lactococcus lactis and Streptococcus thermophilus. But you also have other bacteria that can play a role on the fermentation process, which are non-starter bacteria, but also some secondary microbiota in which Brevibacterium are part. So each microorganism or bacteria that will grow in the fermented food will bring unique flavors and textures to that product. So when we consider the context of food fermentation, so if you have bacteria, you will also have bacteriophage. So wherever bacteria thrive, so you will find bacteriophages. So what is that? So what are phages? So phages are the viruses that infect bacteria. They are considered the most abundant biological entities in our planet, and they outnumber bacterial cells in environmental samples. Phages are very, very diverse. So you can see different shapes, so that it's given by the viral particle. So here, for example, you can see so phages with long capsids, other with smaller, some phages with tails, some phages that are more like filamentous, some that don't have any tails. And phages also can have different genetic materials, which can be DNA or RNA, and can be single-stranded, double-stranded RNA. But the most abundant phages that we know so far is the tailed phages with double-stranded DNA. So why are phages important in the context of food fermentation? So first of all, because phages are part of the dairy ecosystem. So it's the most, in the context of food, the dairy environment is the one that we know the most, is the one that we have been studying the most. So we have several studies that show the presence of phages in the cheese or in the dairy facilities, in the whey powders. So, for example, when the dairy industry use the whey as in the fermentation process, so the dairy is also bringing back some phages in those samples, and the phages in whey powder also has been shown to be very heat resistant. So here, for example, we have a study that it was published last year, that it's a result of a longitudinal study, like from 20 years of phage testing and isolation in a dairy industry that it's partner of the lab where I work. And they isolated more than 200 unique lactococcal phages, which was characterized by MLST. And for you to have an idea, one of the phages that it was called GL7 was isolated 132 times throughout this 20 years. So if you have a high population of bacteria growing in an exponential phase, you are also going to find phages. And you have here examples of phages infecting several different bacteria that are important in the context of food production. So you have like starter cultures, non-starter cultures, you have probiotics, so you have wine related LAB. So yeah, phages are everywhere in this context. And so that being said, so phages in summary can be our friends or they can be our enemies. So they are our enemies when we are talking about phages that infect LAB or ripening strains, because they can block and slow down the bacterial fermentations and generate low quality products. But they can also be our friends when they infect pathogenic and spoilage bacteria. So because they are able to control bacterial population, they play a role on nutrient cycling, and they can be used also in biocontrol. We just heard this great talk from Thiago, and so phages has been explored nowadays as an alternative for against pathogenic bacteria that are resistant to antibiotics in the context of phage therapy, which is not going to be the goal of this talk. But yeah, phages are also being considered some of our friends. And here you have some examples of some commercially available phage products that you can find that it has been approved to be used. So with that, considering that phages can be our enemies and that they can infect LAB and they infect ripening strains, so it's very important that we understand the interactions that phages have with their host bacteria, because with that, we can improve the quality of fermented food by protecting strains against phage attack. So what is that? What is phage-host interactions? So phage-host interactions, first of all, refers to the dynamic and complex relationships that phages will have with their bacterial hosts. This involves how phage infect the host, so how bacteria are able to defend themselves, how they are able to resist to phage attack, but also how phage counterattacks. And these interactions will impact several ecosystems, and including those related to food production. I will not be focused on how phage counterattacks, but there are several strategies related to it. And we recently published a review on that subject in the context of lactic acid bacteria. So if you want to know more about it, you can take a look. And so when we consider the phage infection cycles, there are two main cycles, which are lytic cycle and the lysogenic. So in the lytic cycle, you will have the bacteria. So you will have the phage adsorbing to the bacterial host, and then they will inject the genome inside the cell. And so phages will take control of the bacterial metabolic machinery, and then it will replicate the genetic material with most common DNA. And then the RNA is going to be transcribed, the proteins are going to be translated, and then the genome will be packed inside the phage capsid. The virions will be assembled, and after that, the phage will be released from the bacterial cells. So this is a very coordinated movement. So for some dairy phages, this cycle takes about 30 minutes, and every 30 minutes, each cell can release about 100 new phages. This can vary depending of the cell. So yeah, so some phages will have less particles, some will have more. But yeah, in average, for some well-known dairy bacteria, will release 100 new phages per cell. And then for you to have an idea, so you have one bacterial cell. In the doubling time, you will have 2 new cells, and then it will double. You will have 4, then 8, and so on. But for phages, like after you have one phage, after 30 minutes of infection, you will have 100 new phages, and then you have 10,000 new phages, and then you have 1 million. So, and that's or started with 1 phages, so that will be ready to infect other nearby cells. But so also it's important to say that the phages that are able to enter and they only do the lytic cycle are what we called lytic or virulent phages. And on the other hand, if the phage can go through the lysogenic and the lytic cycle to one on the other, so it's called temperate phages. When the phage genome is integrated in the bacterial genome, it's called a prophage. So for the prophage, what happens is that for the temperate phages, what happens is that the phage absorb and then they inject the DNA as the normal cycle, but and then instead of replicating the DNA, the genome of the phage recombine in the genome of the bacterial host, and then integrates in the bacterial host until, and it will be replicating with the bacterial host until there is a signal that will generate the genome excision, and then after the genome excision, phages will enter again the replication cycle, and then they will be able to continue the lytic cycle and spread new virions to the nearby cells. So how do bacteria defend against phages? So now we understand an overview of the phages life cycles. So how do bacteria defend against phages? So there are 2 main strategies. One of them is the mutation of host factors. So bacteria will, you will have some subpopulation of bacteria that have mutations in the proteins or the structures that will interact with the phages. So in the case of the adsorption, the bacteria will have some mutations in the phage receptors, and then phages are not going to be able to absorb or to infect. And so you have also some mutations in factors, in host factors that are related to the genome injection that also can stop the bacterial, the phage genome injection. But there are also the defense systems. So nowadays we know several different defense systems. We have EBI, we have BREX, we have Disarm, and several others. But also we have those that it's very well known, which is the restriction modification system and the CRISPR-Cas that are used to cut the phage DNA upon infection. And if you are curious, for example, to know if the bacteria that you work with has phage defense systems, you can use so some of tools that have been published recently, like Padlock or Defense Finder, and then you can see and you can analyze your bacteria. And recently, there is been published a review with all the most recently found anti-phage defense systems of bacteria. So when we consider, for example, the mutations of host factors and the block of phage absorption, so so this is the first line of defense, and we are going to see a little bit about it with the example of Lactococcus lactis. So Lactococcus lactis, as you probably know, it's a key microorganism in the dairy industry. So it's used in the production of several cheeses. Here in Canada, it's very important in the production of cheddar. It's used in the production of buttermilk and several other fermented dairy products. So as other lactic acid bacteria, it converts lactose into lactic acid and will contribute to acidification and the preservation of dairy products. And so how can we protect industrial strains from phages? So when we talk about the context of starter strains, you can do three things mainly. So one of them is to rotate the starter cultures. Another thing that you can do is change the bacterial strains. And the third one is to modify the bacterial strain. So like, if you remember the study that I showed that it was done in partnership between the lab where I work and the dairy industry, they monitored the phage population and they studied the phage population in the dairy industry during a time of 20 years. And then when you see a population, when you do this type of work and you see that a phage population it's increasing, you can remove the bacterial strain from the use in the dairy fermentation, or you change your rotation scheme of the starter cultures. But so I will focus on the modification of the bacterial strains. So, and one way to do that is by selecting bacteriophage insensitive mutants, which we call BIMs, and that's how I'll be calling them from now on. So what are BIMs? BIMs are a natural subpopulation with mutation in key genes involved in phage infections. So in other words, they are phage resistant, and we can select them among the population of sensitive strains. So here you have an example of how we do the experiments for the selection of BIMs. So you have your bacterial loam here, no phage whatsoever, but and then you will start adding phages, you start seeing the formation of plaques. And if you increase the titer of the phages that you are challenging the bacteria with, you are going to see more phage plaques and almost no bacterial loam. And at the end, when you have a high concentration of phages, you will not see the bacterial loam at all, but you will start seeing some colonies that will appear, and those colonies are BIMs. So they are resistant to phages, and we usually take those colonies and we characterize what is the strategy being used by the bacteria to defend against phages. So in the case of Lactococcus lactis, so the cells of lactis, they are covered by a polysaccharide pellicle, and that differentiates some strains from the others. So, and this pellicle, polysaccharide pellicle, is used by some of the lactis phages to infect the bacterial cells. And one example of these phages is the phage P2. So P2 and the structure that is responsible for the recognition of the receptor in the bacterial strain, which is in this case is the pellicle, is the RBP, which stands for receptor binding protein. So in the case of phage P2, so it can bind to the pellicle in a specific way. But when the RBP is linked here to the pellicle, and then it forms a specific binding, and when you have calcium chloride when you have calcium, you have the change in conformation of the RBP, and then after that you have the DNA injection into the cell. And that pellicle, the differences in the pellicle of different lactase strains will also determine the phage-host range, which is the ability of the phages to infect the bacterial cells. So here you have several different phages, and here you have several bacterial strains. So when you have dark blue, you have resistant strains. When you have light blue, you have sensitive strains. So you can see, for example, that these phages, they are very specific. And for example, here, the 3 less phages, they only infect one strain, each one of them. So that's why rotating the starter cultures is an efficient way to get rid of these phages. And so, yeah, and also when you do the generation of beans with mutations in the phage receptors, you also going to impede, like you are going to remove that ability of phages to bind to the bacterial receptor, and then that strain will be resistant to the phage. So another way that bacteria can defend against phages, as I mentioned, is through the defense systems. And one example is the CRISPR-Cas systems, and that is used, that cuts the genome of the phage. And one very well-known example is the Streptococcus thermophilus. So Streptococcus thermophilus is also a starter strain. So it's used in yogurt and many cheeses production around the world. It's carefully selected, so they have very desirable technological properties that it's linked to the flavor and the texture of the products where they have been used. And there is an estimative that the humans consume annually around 10 to the 21st cells of Streptococcus thermophilus. Nowadays, we know about 5 different genera of Streptococcus phages. So they are all virulent. There is no knowledge of temperate phages, and they are double-strand DNA phages genomes, and then they have around 30 kb. So they are not, they don't have very big genomes. And here you have the examples of the 5 different groups that you have characterized so far. And a few years back, the lab participated in the characterization of the CRISPR mechanism of the CRISPR mechanism of action. So some work was done and showed that CRISPR was able to provide resistance against viruses in bacteria. And then this work was done in partnership between Université Laval and Danisco Dupont, and it was done in Thermophilus. And later they showed also that the CRISPR-Cas system in bacteria were able to cleave the bacterial and plasmid DNA. So what CRISPR stands for? I'm sure that by now all of you know, but CRISPR means clustered, regularly interspaced short palindromic repeats. And they are, the CRISPR locus, they are associated with some proteins which are the Cas proteins which stands for CRISPR associated. So if you have a sequence of DNA there, like you can see just a bunch of letter, and then if you highlight, you are going to see some patterns which are the repeats. So they are clustered, so they are together. They are these repeats, they are regularly interspaced. So between them, you can find some sequences that are what we now know they spacers, and they are short and they form palindromes. So you have nowadays also several different CRISPR-Cas systems characterized, but the one from Streptococcus thermophilus is the type 2A, which has a Cas9 protein and other proteins that it's Cas1, Cas2, and CSN2. And so there is a difference between what we call the CRISPR-Cas system as an adaptive immune system of bacteria and the Cas9 2. So the 2, you remove some components of the system and then you put in a plasmid, and then you use whole genome engineering. That's not what we are talking. We are talking about the adaptive immune system of Streptococcus thermophilus. So, and how the CRISPR-Cas system is used to defend against phages. So the first step is the adaptation or acquisition. So you have a phage that will infect the bacterial cell and it will inject the DNA. So in the first encounter of the bacterial, of that bacteria with that phage, so the Cas1 and Cas2 will bind to the DNA of the phage in a very specific region, and then it will cut the DNA of the phage and it will insert that fragment into the genome of the bacteria in the CRISPR locus. And then you later you have the expression stage in which the tracer RNA and the CRISPR locus will be transcribed in the pre-CRISPR RNA, and then they will be cut and processed in this small guide RNA that will bind to Cas9, for example, and that same phage that phage infect the bacteria again. So Cas9 and this surveillance complex will interfere. It will perform the interference stage, and so they will bind to that DNA. It will create a double breaks, and then the phage will be inactivated. So you can also select for beans in Streptococcus thermophilus, the CRISPR beans, and the process is the same. You are going to challenge the bacteria with the phage, and then you are going to recover some colonies that will be resistant to the phage. And so you can see the acquisition of a new spacer when you do a PCR of that colony in the CRISPR array, and then you are going to see here you have the wild type, and then in the one that you challenge with the phage, you will see a shift in the band pattern on the PCR gel, that it will be 66 base pair in the case of Thermophilus, that it's related to 36 base pair repeat plus the 30 base pair spacer. So, and then that new spacer will be added in the 5' end of the CRISPR array, and it will generate. So here you have the strain that is sensitive to some phage. Here you have the spot test on the bacteria alone, and then caused by the sensitivity to that bacteria to this phage here, 858. But and then for the phage 2972, which was the bacteria was exposed to and acquire a spacer against, so the bacteria is now resistant to that phage. So what the dairy industry can do is to use different phages. So in the case that they have a collection, or they have partnerships of labs that have a collection of phages, what they can do is that they can expose their beloved strains to the phages. And then, so you have here, for example, the white type strain, and then you expose that strain to one phage. And then you see here a yellow spacer being acquired, and then you expose again to a second phage, that it's the blue phage here, and then you have another spacer being acquired, and you continue doing that until the end. You have some starter strains that are resistant to several different phages. And for those that want to introduce some studies related to CRISPR in their classroom, in their courses at the university. So this has been taught in the university since 2012, and in 2017 the lab published the protocol for to evaluate the CRISPR-Cas acquisition in the classroom settings. So nowadays, I've been working with the CRISPR targeting, but in my case, I'm using more in the context of genome engineering. So I am working on the publication of an article. So if you are interested about that, so stay tuned, that it's gonna come out sometime soon. So we have been learning about phage-host interaction in the context of starter strains, but there are also other strains that are harder to replace, and so that makes me ask the questions: how to replace what is in theory irreplaceable? So our knowledge about phage infecting starter bacteria it's very broad, and so we have several phages, phage genomics being studied in starter strains. So Thermophilus, in Leuconostoc, we have in Lactis, in several other bacteria. But also we have a limited knowledge on phages infecting ripening strain. And in that context, my previous work was related to phages in Brevibacterium aurantiacum, which is a very important bacteria used in the production of washed rind cheeses that it's produced around the world. This is a gram-positive bacteria that it's tolerant to high concentrations of salt, and you can see here this carotenoid pigments being produced by the bacteria, very orange, bright, and very beautiful. And this gives the color of the rind of several cheeses produced around the world. In my case, I study a strain that it was called SMQ 1335, which was an industrial strain, and that it has been for many, many years commercialized and built as B. linens, Brevibacterium linens, and many of the strains that it's commercialized as Brevibacterium linens, it's in fact Brevibacterium aurantiacum. So this SMQ 1335 was used in the production of washed rind cheeses here in Canada, and it was linked to the flavor and the color of the cheeses that they were used. And we cannot rotate a ripening strain, and it's very, very hard to replace. However, this my project is started because this strain, they were having trouble in the production of the cheeses, and we later saw that they were sensitive, this strain was sensitive to phages. So, and then we isolated phages from the three manufacturing sites that they had. So we, in total, we had 62 phage isolates, and we sent 16 of them for phage genome sequencing. They were isolated from different places inside the manufacturing sites. And then we studied the genomic of these phages. So later we also we used one of those phages to see what it was the impact that they have in washed rind cheeses. And you can see here the control of cheeses with no phages that you can, and here the other cheeses with that we added the phages. So from day 13 it's even more clear, and from the following days you can see also a very big difference between the ones that we didn't add any phages and the one that we add phages in the development of the color of the rind. So if we cannot rotate and we cannot replace, or it's going to be very hard to replace, how can we protect this strain from phage predation? So, and the answer, I'm sure you all know by now. So it's the generation of beans. So we performed 2 different experiments in liquid and in solid, in which we exposed the bacteria to the phages, and then we had several colonies that we took and we characterized. So we performed Illumina sequencing of 11 beans, and also we took the white type strain that we used to generate the beans, and we also performed Illumina sequencing of that, and we compare the reads. We aligned the reads with the previous sequence of PacBio that we had from a few years back in 2016 of the same strain. And then we did the mutation mapping of the difference compared to the wild type genome. And we found that 3 of the beans that we selected, they had mutations in the same system. It was the ABC transporter, and here, so you can see that these phages, they were completely resistant to the phages. So there is no plaques there, but the bacteria, the beans, were completely resistant to the phages. But the phages were still able to adsorb to the bacterial cells, so it was not related to the receptor, but another mechanism after the adsorption. So these were fully resistant, and when we analyzed the genome of the beans, we saw that each one of them, these 3 beans that we selected, they have mutations in some components of the ABC transport system. So bean 4 had in the substrate binding protein, and then bean 2 and bean 6, they had the mutations in the permease, which completely inactivated the proteins. So we have frame shift or nonsense mutations that completely inactivated the protein. Unfortunately, this strain, Brevibacterium in general, it's very, very hard to have protocols for genetic transformation. And it's not that we haven't tried, we have tried, but it's very, very hard. So we cannot confirm with the genetic studies the role of this these systems in the resistance of phages, but we have a good indication that yes, with the mutations in this three components of the system, in the same system giving full resistance to the phages, we have a good indication that this systems is somehow involved in the interaction of the phages with Brevibacterium aurantiacum. So, so to conclude, what I want you to keep in mind as we finish this talk is that the phages are part of the dairy ecosystem. They are there, and if you have a sensitive strain, they will replicate very, very rapidly. But only know that they are present is not enough. So we can design strategies to protect industrial strains from phage predation. And when we study phage-host interactions, we can select for phage-resistant mutants, and also we can understand better how bacteria can defend against phages. And further away, you can also understand how phages counterattack those defense systems of bacteria. And with that, I'd like to thank you all for your attention. I'd like also, yeah, to thank all the people that participated in during my academic career. And if you have any questions, I'll be glad to take any questions that you might have. Thank you.
Thank you very much. That's great. Can you hear us?
Yes.
Yes. Thank you so much, Alessandra. It's a great talk, full of amazing experiments. We have some questions from the audience. I just have a curiosity before the questions.
Yes.
Does your Brevibacterium strain have a CRISPR system?
No.
No. Okay.
No. Yeah.
And another stupid question maybe, but if it had and it had acquired a spacer, would it be considered a genetic mutant, a GMO?
No. No. So that's what here in Canada we are not allowed to have GMOs in the food industry. So and this is a very natural way to select. So you are just selecting for some subpopulation that acquired resistance, and that could have, you are forcing in the lab with the exposure of the bacteria to the phages, but it could happen in nature if the phage had encountered the bacteria naturally. So you have like when you PCR the CRISPR, or you do the genome sequencing and you check the CRISPR loci of different bacteria, you can see that it acquired resistance to, it has a spacer against several different phages, which mark the encounter of that bacteria to the phages in the natural places where they have been. So yeah, no, they are not considered GMO.
On the other hand, if we take the strain to the lab and add the CRISPRs ourselves or the spacers ourselves, because one could do that, then it would be a GMO because you made it out of PCR and stuff like that. Because you could do that too, I imagine.
Yeah. What also we can do is like, but it's not a GMO as well, because like we can force the CRISPR to take some fragments that we want. So, and we do that because we put the PAM, that it's that sequence that it's recognized by the CRISPR-Cas system. So we put the PAM and the spacer in a plasmid. So when you put and then the CRISPR-Cas system will acquire the spacer from that plasmid, and then later you perform some plasmid loss experiments. So you remove the plasmid, and then you will have a bacteria that you force the spacer that you wanted, but it's not a GMO because you removed the plasmid at the end.
Brasileiro, but great.
Yeah.
We have some questions here. Icaro, and then I'll ask Slav's question. How do environmental factors such as temperature and pH influence the effectiveness of bacteriophages in fermented foods, especially in traditional fermented foods?
So does he mean in the context of control?
I think, like in controlling pathogens, I imagine.
Yeah, so I'm not very familiar with the use of phages as control in food. I study them more as our enemies. So in the context of infecting LAB, but we have seen that the pH and the temperature can affect the viability of phages. In some cases, for example, the whey protein, they can the whey products, they can protect the phage from inactivation by heat. So it's something that in that case that the industry wants to use the whey products in the fermentation, they have to use other strategies to remove the phages and not heat, because they can bring back some phages to the production.
Yeah. And I think like in the context that he asked, one thing that it's hard to use phages is because the phage has to encounter the bacterium, and the phage doesn't move. And if it's like in a solid surface, like in a cheese, you're gonna put the phage in a place. It could be right next to the bacterium, but if it doesn't touch the bacterium, then it's not gonna kill the bacterium. So the effectiveness is affected by contact, because you might not have it.
And I think also the abundance, in this case, it's the abundance of the bacteria you want to encounter, because in our case that with preview bacteria, we also had the solid matrix. It was the cheese rind, and we still saw the problem caused by the presence of phages encountering sensitive strains. So it affects the color of the rind. But in that case, the bacteria was very abundant because we were washing the rind of the cheese with the bacteria, and then but in the case of a contaminant, yes, this can be the encounter of the bacteria and the phage can be a problem indeed.
I see. Yep. Slavs asks, are some proteinase inhibitors active against bacteriophages? He means proteinase inhibitors produced by started LAB cultures.
Good questions. And my answer is I don't know. So unfortunately, I don't know the answer for that questions. But yeah, might be, but I don't remember reading anything specifically about it.
Well, some of the other groups of bacteria. They can produce the protease inhibitors against viruses. And some of the lactic acid bacteria, in fact, they are producing some work showing that some enterococci, they can produce protease inhibitors with activity against some viruses. But well, bacteriophage is a virus as well. So I was thinking that you maybe have some more information of this, but most probably they are. So just need to be rapid and discover.
Yeah, exactly.
All right. Any more questions from the audience here? I think everybody's hungry. It's lunchtime. Alessandra, thank you so much.
Thank you very much, Uelinton, for the invitation.
So the morning section is over, and we'll see you again at 1 p.m. Brazilian time. So take care, guys. Hi, Natalie. Can you hear me?
Yeah, I hear you. Hello. Thank you.
Hello.
And you hear me also?
Volume is a little bit too low. I'll just wait.
Okay. I will try to speak next to the microphone.
Can you say something?
Yeah. You hear me?
Yes, it's not too loud though.
Whoa. I will try to speak more. I like this. It's okay?
Okay. You have Marianne also with you?
Yeah. Yeah. Hi.
All right.
Hi everyone.
Hi.
Do you hear me? Do you hear me?
A little bit. Yeah.
Oh, I don't know what is the problem.
We'll try to tune the volume up here in the auditorium to see.
Okay. Okay.
Let's see if it's... No, it's not here. Well, all right. Good afternoon, everyone. Welcome back to our course. I have now the pleasure to introduce you Professor Nathalie Conneau. She's going to be talking all the way from France, University of Rouen. And the topic is Enterococcus, Pediococcus, Probiotics in their interactions with the host. Professor Nathalie Conneau obtained her PhD in microbiology at the University of Nantes, and the prize, that's going to be hard for me, Jean Chauchot Bernard Beffreux, from Nutrition French Institute for the project Regulation of Intestinal Permeability by Probiotics in 2003. Between 2003 and 2006, she was a postdoc in various laboratories and research centers. In 2006, she was recruited as associate professor in microbiology and cell biology at the University of Rouen, Normandy University, France. She's now full professor since September 2022. Her research interests focus on probiotics, gut microbiota, bacterial pathogens, antimicrobial peptides, bacterial-host interactions, inflammatory bowel disease, and digestive cancer. She headed and co-headed 6 PhD students from France, Tunisia, and Italy, and co-authored 49 publications in peer-reviewed journals. She's editorial board member for Frontiers in Nutrition since 2022. Nathalie, thank you so much for your presence in our winter course, and I leave the floor to you.
Thank you. I couldn't see the presentation, please.
Yeah, Ana, podem colocar a apresentação, por favor?
Oh, yeah.
Just setting it up.
Okay.
Yeah.
Yeah, it's okay. Yeah.
The volume is still low. Ainda está baixo o volume. Se der para dar uma aumentada. Tá. Okay. Thank you, Nathalie and Marianne. It's with you now.
Thank you. So thank you for inviting me to this winter school in this microbial interactions. I will talk about Enterococcus, Pediococcus as probiotics and their interactions with the host. I'm a professor in the laboratory of University of Rouen, and my PhD students will also participate in this conference. How to change? Okay. So the genus Enterococcus. So there are some bacteria gram-positive cocci, initially described as streptococci until 1980s. Their fecal streptococci are grouped in the genus Enterococcus, and other streptococci are reclassified in lactobacillus and streptococcus. Enterococcus is the third largest genus of lactic acid bacteria after lactobacillus, which has changed recently in several genus, and streptococcus. Most enterococcal species can grow in harsh conditions. in the presence, for example, of 6.5% chlorous sodium and 30% bile salts, and grow IPH like 9.6. A lot of species can also grow at temperatures ranging from 10 to 45 degrees. They can survive, for example, 30 minutes at 60 degrees. And the two species, Fecalis and Faecium, are the most important species of Enterococcus. We can find them in medical and food microbiology, and they are studied as potential probiotics. So what about the habitat of this genus Enterococcus? Enterococci occur in many different habitats. We can find them in soil, surface water, sewage, plants, gastrointestinal tract of humans and also animals. They are present in foods of animal origins. We can find them in fermented and cooked meats, for example, or also in cheese. Enterococcus fecalis is a predominant Enterococcus species of the human gastrointestinal tract, but sometimes in some countries or in some individuals, it's Enterococcus faecium which predominates. Here you have some examples of colony-forming units we can find in feces, 10^5 to 10^7 for fecalis and 10^4, 10^5 for faecium. Enterococci are also present in the intestine of animals, and this leads to contamination of milk and meat. They can survive pasteurization due to their heat resistance, and cross-contamination can occur at the final stages of processing during slicing and packaging. What about Enterococcus in the food technology? They are used till long time as dairy starter culture. They have major role in improving flavor and quality of cheese. They contribute to the ripening and aroma due to proteolytic and steatolytic activities, as well as the production of diacetyl and volatile compounds. They also produce many bacteriocins called enterocins. As their name is Enterococcus, the bacteriocin will be called enterocins. We have, for example, enterocin A, B, P. Remember that these bacteriocins are small peptides with antimicrobial activity towards closely related gram-positive bacteria, including spoilage or pathogenic bacteria such as Listeria. So we can use the bacteriocin or bacteriocin-producing enterococci as starter culture or protective cultures, or direct addition of the enterocins in food fermentation process, and this may help to prevent the growth of pathogens. Enterococcus also exists as probiotics, and here are some examples. We have the Enterococcus faecium SF68, found in Silactin product and biofluorine. It is used for treatment of diarrhea, and it is considered as an alternative to antibiotic treatment. We have the Symbioflor 1 from the firm Symbiofarm. It contains Enterococcus fecalis, used for recurrent infections of the upper and lower respiratory tracts, inflammation, for example, of the nasal sinuses and bronchi. And also we have some products for animals, like Symbiopet. Symbiopet is a nutritional supplement for dogs and cats that contains Enterococcus faecium. It can help stabilize and reinforce the intestinal flora of the pets. Now some information about the pathogenicity of Enterococcus. Enterococcus belong to lactic acid bacteria, but there is controversy of this bacteria, considering them as generally recognized as safe. In fact, in the last decades, they have emerged as important nosocomial pathogens causing a variety of infections in immunosuppressed and intensive care unit patients, with infection like endocarditis, bacteremia, infections of the urinary tract. We found that Enterococcus fecalis predominates among Enterococci isolated from human infection. More than 80% of the strains are strains of fecalis in these infections. Several virulence factors have been described, and the number of antibiotic-resistant Enterococcus, especially vancomycin-resistant Enterococci, is increasing. The virulence factors of Enterococcus help the microorganisms to cause disease. Here are some examples of virulence factors of Enterococcus fecalis. The virulence factors can be involved, for example, in attachment to host cell and to extracellular matrix proteins. They can help for resistance to macrophages. They can provoke cell and tissue damage, and they can help also the bacteria to evade the immune system. These virulence factors can be present on plasmid genes or on chromosomal regions. Virulence factors on plasmid are transmissible to other strains by gene transfer. And it is known that Enterococcus faecium generally contains less virulence factors than fecalis. So, for safety assessment, before use these bacteria as probiotics, virulence factors must be checked in each new isolate as well as the antibiotic resistance. The antibiotic resistance of Enterococcus is very wide. There is a resistance to a wide variety of antibiotics contributing to pathogenesis. We can find some intrinsic resistance to many antibiotics, which genes located on the chromosome, and we have some acquired resistance from a lot of other antibiotics with genes on plasmids that can be transferred between strains. Vancomycin resistance is of special concern because this antibiotic is considered a last resort for treatment of multiple resistant enterococcal infections. So the emergence of vancomycin-resistant Enterococci in hospitals has led to infections that cannot be treated with conventional antibiotic therapy. So let's go to the experimental part. So with some students from Tunisia, we have isolated some bacteria. We use samples from traditional fermented Tunisian dairy products and also samples from artisanal dried Tunisian meat, which is called dried husband. We have isolated and detected the Enterococcus strains using phenotypic analysis. The identification was performed using multibiotyper and also using specific PCR, as you can see in this electrophoresis. We use a target of specific gene for fecalis and faecium. So in the traditional fermented Tunisian dairy products, we found two strains of Enterococcus fecalis that we have isolated, named OB14 and OB15. And in the Tunisian meat dried husband, we find 5 strains of faecium named MZF1, 2, 3, 4, and 5. So after this, we have characterized, we have characterized the probiotic properties of the strains and compared this to the Enterococcus Symbioflora probiotic reference. So the first study was to look at the tolerance to acid and bile salts because this is what the bacteria need to support when she is present in the gastrointestinal tract. So we found that this isolate were tolerant to acid and bile salt. The fecalis strains isolated from dairy products, but also the husband meat bacteria showed high tolerance to these gastrointestinal conditions. We look at the autoaggregation capability because this is a criterion for selection of probiotics that may help the bacteria to adhere to gut. So we measure this after 24 hours, and we found among these strains that faecium MZF1 and 2 had the high autoaggregation, which was superior to Symbioflora 1, the probiotic strains known. We look after the adhesion to intestinal cells using the model KECO2-TC7 intestinal cells, and adhesion has been performed after 3 hours of treatment, and we enumerated the adherent bacteria by plating, and we find that Fecalis OB14, 15, and Ephesium MZF5 had a capacity of adhesion higher than the probiotics embryo floor. After this, we look also at the cytotoxicity using the same models of cells and the measurement of the LDH. So it is an enzyme with its release only after necrosis, lysis of cells, when this is mortality of the cells. And we found no cytotoxicity of these potential Enterococcus probiotics. We can see here that the cells are intact. And after this, we look at the modulation of the intestinal barrier. After overnight incubation of Enterococcus strains with differentiated KECO2-TC7 cells culture on inserts, we measure the transepithelial resistance with an ohm meter. And we find an increase of this transepithelial resistance using these strains, except with one of the strains, the MZF1. So many of these strains had beneficial effect on the intestinal barrier. After this, we wanted to detect if these isolates can produce some bacteriocins. So we look at the more frequent enterocin known, that is enterocin A, B, and P, using the detection by PCR. And we found no detection of these genes in the Fecalis strains. On the contrary, in the Faecium, we found many of these enterocins, and there is a synergy because the antagonism tests show high anti-Listeria activity. Then we look at the virulence factors. In the Fecalis isolated from dairy products and the Faecium isolated from the husband meat. And we found in Fecalis some virulence factors mostly related to adhesion. And in Faecium, there were less of these genes as described also in literature. After this, we look at the antibiotic susceptibility resistance. We find that the Fecalis and Faecium strains were sensitive to ampicillin, vancomycin, and gentamicin, and were resistant to erythromycin, which is interesting resistance in this genus. So in conclusion to this part and perspective, we isolate some Enterococcus from Tunisian products. We identified Enterococcus fecalis and Faecium strains. We characterize their probiotic potential. We find some tolerance to gastrointestinal conditions. We find high capacity of autoaggregation and adhesion. We find that these bacteria were able to modulate the intestinal barrier. We find also that many of these strains can produce some bacteriocins. We find absence of cytotoxicity, and we found antibiotic susceptibility to many antibiotics. So most of these isolates that we found in these Tunisian products of dairy product and meat product possess the principal requirements for probiotic. After this, we need to in vivo invalidation before the use of these strains in food technology. And also we have made the in vitro validation because we have performed the whole genome sequencing of all these strains. So what can we conclude in this part also is that we need to have a case-by-case examination of safety. In fact, it is often necessary for validation of a new Enterococcus strains as probiotic or protective culture to check of the safety of the strains, and the legislation may vary by country, so we need to follow the specific guidances. So let's talk about now the genus Pediococcus, because we have also studied some Pediococcus strains in our work. So Pediococcus is a gram-positive homofermentative bacteria belonging to the lactic acid bacteria group, close to the genus Lactobacillus, you can see here. The genus Pediococcus includes several species. You can see some name here, and the more known are Pediococcus acidilactici and Pediococcus panthosaceus. These bacteria are frequently isolated from various food sources like ripened cheese, beverage, pickles, wine, dairy, and meat products, and they are involved in the manufacturing of fermented foods. These bacteria are also generally recognized as safe. Here you have the example of the species Pediococcus panthosaceus and what are the benefits of these bacteria in technology and for health. So you can see here that this bacteria is involved, for example, in the fermentation of dairy products, fruits and vegetables, meat products, and also pasta. And for the health characteristic of this probiotic, some studies found antioxidant properties, found suppressive to foodborne pathogenic bacteria, so can be used as biopreservative agent, and this is partly due to the production of bacteriocin. Some strains were found to be able to lower the cholesterol, to have some immunomodulatory function, and can be used also maybe to treat cancer. This is an hypothesis because they can kill some cancer cells in vitro. So here I will present you a little part of experiment we conducted also with a collaborator from Tunisia. So we studied Pediococcus strains, which was Pediococcus panthosaceus MZF16, which was isolated also like the Enterococcus strains I presented just before in the meat called dried husband. So we studied the autoaggregation of the bacteria. And also, we look at it on the scanning electron microscopy. So we find high autoaggregation capacity of this strain. And also, we look at the hydrophobicity, as hydrophobicity can help the bacteria to adhere to eukaryotic cells. And we find good hydrophobicity using some various solvents. After this, we look at the interaction of the bacteria with eukaryotic cells using several tests. So the adhesion capacity was good adhesion capacity related to the high autoaggregation we found just before. We see that there is no hemolytic activity here. Also, there are no cytotoxicity towards the model KECO2 TC7 cells, intestinal cells. We found that these bacteria can stimulate the immune system. In fact, we found some secretion of low level of interleukin-8. And also, using differentiated cells, we found that these bacteria can also increase the resistance of the intestinal cells, so improve the intestinal barrier. We look at the antibacterial activity of this strain. Using antagonism test, we found an inhibition zone with Listeria monocytogenes. So this bacteria can produce maybe a compound which is active against this bacteria. So we use a traditional test on proteinase K to show that it is a peptidic activity. So related to these bacteriocins, because it was lost when we use this enzyme. And we use PCR to detect the pediocin genes in the strains. So here you have the sequences of these pediocins from strains, and we have the sequence also of the immunity protein. We studied this using BLAST-X and compare with the bacteria species Pediococcus acidilactici. And we found that Pedioxin MZF16 contains the motif responsible for the anti-listeria activity here, and also the 2 disulfide bonds characteristic of class 2A bacteriocins. And also we found a mismatch at the C-terminus of the peptide. When we compare the 2 bacteriocin, Pedioxin, Panthosaceus, and the acidilacticine, the threonine residue was present in Pedioxin instead of an asparagine. And this sequence we found in strains was totally identical to another bacteriocin, which is coagulin, a Pedioxin-like inhibitory substance produced by Bacillus coagulans. So in conclusion to this part, we have isolated new strains, Pediococcus MZF16, from the host-bound Tunisian products. We characterize the probiotic potential of this bacteria. We found that this bacteria is tolerant to gastrointestinal conditions. We found that this bacteria has high capacity of autoaggregation and adhesion to intestinal cells. We found that this strain can modulate the intestinal barrier and also produce some postbiotics, these bacteriocins. Pedioxin, which we see it's similar to coagulin A. We found an absence of cytotoxicity, and we found that this bacteria was sensitive to antibiotic. We also performed some in vitro validation with totally sequencing the genome of these strains. So according to these results, Pediococcus panthosaceus MZF16 can be proposed as a probiotic and bioprotective agent for fermented foods, including Tunisian dry meat and sausages. And further investigation will aim to study the behavior of this strain in meat products as a component of functional food. So here I want to acknowledge all the members of the lab participate in this study, and my collaborators from Tunisia and Germany, and also the 2 students who conducted this experiment, Alpha Bakouri and Mohamed Zoumiti. Here are the work we have published with all these experiments. I want to thank the Winter School for inviting me in this session of microbial interaction, and thanks to the Professor Svetoslav Todorov. And for the following, we have talked about the bacteria. We have talked about the interaction between the bacteria and the cells, the effect of the bacteria on the cells. But what about the effect of the cells on bacteria? What about, for example, the human stress and the hormones that are produced and their effect on bacteria? And for this, my PhD will continue the presentation. So Miriam Boujnan.
Hi, everyone. So my name is Miriam Boujnan. I'm a second-year PhD student in microbiology, supervised by Professor Nathalie Cunil. So we will talk in this second part of our presentation on Enterococcus, Pediococcus as probiotics, and also and their interactions with the host. So first of all, before starting, I would like to thank Professor Slavis Todorov for this invitation, and also my supervisor to allow me to present with her part of our research work in the laboratory. So we all know that the gut microbiota is the largest microbiota in the human body. It's such a rich and diverse ecosystem, and it plays a very important role in the human health due to its several functions, including protection against pathogen colonization, regulation of immune system, metabolism, host physiology, and mental and neurological functions. So it becomes now an evidence that the microbiota or the gut microbiota communicate with the host, and it's such a dialogue or bidirectional communication between both of them, which is established along what we call now the microbiota gut-brain axis. So the microbiota and the host communicate with each other via various routes, including the secretion and recognition of microbial metabolites, and also host neurotransmitters, like, for example, stress hormones, including adrenaline and noradrenaline, which are also called epinephrine and norepinephrine. So these two molecules belong to a family called catecholamines, thanks to their structure, and they are both neurotransmitters and also stress-related hormones. So the release of this molecule in the human body impact not only the host physiology, but also its microbiota. And there is a recent field of research called microbial endocrinology, and there are several studies that have demonstrated that catecholamines, including norepinephrine and epinephrine, can modulate bacterial physiology, especially their growth, violence, adhesion, and biofilm formation. So that was explained by the fact that this catecholamine can be, on one hand, can play a role of siderophores, or this response can be explained by the fact that these bacteria can have a bacterial adrenergic sensor. So till now, there are a lot of studies on catecholamine effects on bacteria, but especially on pathogenic strains. But we don't have too much data on beneficial or probiotic bacteria. So the aim of my presentation is to do an overview or summary of current literature and also our research work in the laboratory on catecholamine effect on some probiotic strains, especially how they modulate the probiotic physiology, their interactions with the host, and also the probiotic and pathogens host interactions. So in our laboratory, there are many studies conducted by Professor Natalie Cunil on epinephrine and norepinephrine on some Enterococcus and Pediococcus species, including Enterococcus faecalis and Enterococcus faecium. And now I'm currently working on this strain, which is Pediococcus pontosaceus. So the first test was the autoaggregation assay. We all know that the autoaggregation is a crucial step. It's very important criteria within probiotics, which allows facilitates and allows the adhesion of these probiotics to the host. So we have found that norepinephrine has increased the autoaggregation of Enterococcus faecium and also the autoaggregation of Pediococcus pontosaceus. So we were curious to visualize this phenomenon using scanning electronic microscopy. So we have, as you can see here, there is the autoaggregation of Pediococcus pontosaceus, but when we treat it with norepinephrine, we can see in these pictures that there is more aggregate, and interestingly, we have observed that there is potentially more secretion of exopolysaccharides. In parallel, we have also investigated the effect of catecholamines on probiotic strains biofilm, because biofilms are also important to the bacterial colonization of the host. So for all tested strains, notably Enterococcus faecalis, Enterococcus faecium, and also Pediococcus pontosaceus, we have noted that catecholamines, epinephrine and norepinephrine, promoted biofilm formation within these strains. Regarding bacteria and the host interaction, we were investigating how these molecules can modulate this interaction. So we were interested especially to bacterial adhesion to intestinal cells, CACO2. So for Enterococcus faecalis, Enterococcus faecium, and Pediococcus pontiaceus, we have observed that norepinephrine can stimulate twofold bacterial adhesion to these cells, which is a very interesting result. In parallel, we have also, we were wondering how catecholamines can, for example, modulate the interaction between two bacteria, especially when with probiotic and pathogenic strains. So for that, we have tested two couples of bacteria. The first couple is the probiotic Enterococcus faecium with the pathogen Listeria monocytogenes. And the second couple is Pediococcus pontiaceus with the pathogen Pseudomonas aeruginosa. And we have investigated the role or the effect of norepinephrine or epinephrine on their coaggregation, their adhesion to intestinal cells, biotransformation, and cytotoxicity. So for the coaggregation assay, the coaggregation, it's known that it's an important phenomenon for probiotics to prevent pathogen colonization of the host. So we have observed in vitro that there is an increase of the coaggregation of both couples with norepinephrine. And once again, we were curious to see how they coaggregate together. So for Pseudomonas aeruginosa and Pediococcus pontiaceus, we can see that they coaggregate this way. But interestingly, when we treat the suspension or the co-culture with norepinephrine, there is a different distribution. And also, when we zoom on the picture, we have observed that they are more coaggregate and also a kind of exopolysaccharide layer. Concerning dual species biofilm formation, we have studied the mixed biofilm of both of the Pseudomonas aeruginosa, tagged with green with the JFP protein, and the probiotic Pediococcus tagged with M-cherry protein in red. So we have observed that there is a dominance of the probiotic strain in the mixed biofilm, and the presence of norepinephrine has promoted this dominance. Regarding probiotic pathogen and host interaction, we have performed a competitive exclusion assay by treating our CACO2 cells with cultures of both couples, treated or not with norepinephrine, and we have observed that there is an increase of the adhesion of probiotic strain, but there is the important decrease of pathogens to the cells in presence of norepinephrine. Similarly, we have investigated bacterial cytotoxicity with using the LDH cytotoxicity for measurement of lactate dehydrogenase, and we have observed that norepinephrine for the first couple, Pseudomonas and Pediococcus, norepinephrine promotes the endocytotoxicity of the probiotic of the pathogen. And we didn't see the same effects on the other couple. So to recapitulate, in summary, we have demonstrated that probiotic strains can also sense and respond to catecholamines, and these molecules can modulate, on one hand, probiotic physiology with the stimulation of their aggregation and biotransformation, and also can modulate interaction between probiotic and host by stimulating by increasing their cell adhesion, and also they modulate the probiotic pathogen-host interaction by stimulating co-aggregation and promoting the dominance of probiotic in the mixed biofilms. So we also, in our laboratory, there are also another studies which were conducted by Professor Nathalie Connell on other eukaryotic molecules or hormones such as serotonin and substance P, which modulate also the physiology of the same probiotic strains. So in conclusion, and as a research prospect, we think that future studies on probiotic and host molecules are much needed for a better comprehension of bacteria and host interaction. So thank you so much for listening, and we are here to answer to your question. Thank you so much.
Hi. Thank you both for the very nice presentations. I open now the floor for questions, if we have any questions from the audience. Just a quick second.
Hello, very interesting research. Congratulations. And just a commentary. These neurotransmitters, epinephrine and norepinephrine, are also known as quorum sensing autoinducer molecules. We work with quorum sensing inhibition, and these neurotransmitters are also known as autoinducer molecules.
Yeah.
Yeah, they serve as communication cues in Salmonella. I think it's the group from Vanessa Esperandio in the US. They have shown that. So.
Exactly. Yeah, there is a lot of area to explore. Mm-hmm.
Yeah, interkingdom signaling. That's what it's called.
Exactly.
So I was a little bit distracted. I don't know if I missed something from Miriam's part. So you show the two pathogens together, and then you added the compounds, and you saw there was increased aggregation, and the dominance of the lactic acid bacterium. But what about in the absence of the compound? Isn't it the same? Like, wouldn't you have more dominance of Pediococcus and Enterococcus over the pathogens?
Regarding the dominance is the same, but there is a little, very few difference between treated and non-treated conditions. But we have, by quantifying the biomass and average thickness of the mixed biofilm, we have observed that there is a very slight simulation of both, but it doesn't change the distribution of each strain in the mixed biofilm. There is also always the same dominance of Pediococcus pontiaceus.
I see. Have you done these experiments in anaerobic conditions to mimic the gut?
No, because Pseudomonas aeruginosa, it doesn't support the anaerobic conditions, and Pediococcus pontiaceus is anaerobic bacteria. So we try to find conditions which allow us to do a co-culture of both bacteria in optimal conditions.
Yeah, but I mean, because these compounds are found in the gut, right? So maybe in the other experiments which don't involve the pathogens, if you use anaerobic conditions, would you find different results, maybe even more pronounced results, more coaggregation, more of whatever you're looking for? I don't know if you have thought about it.
Maybe it can change something, but also there are some publications that says that there is some oxygen in a small ecological niche close to the eukaryotic cells. So we have to measure. Yes, I don't know exactly if we have less oxygen as we use, but yes, it will be interesting to test another condition.
Yeah. The only reason I am asking is because with Salmonella, a group from researchers that I know, which are my friends, they tried in aerobic conditions and they put the molecule they're testing, there's nothing. And then when they performed the experiments in anaerobic conditions, there was a huge effect. Like the strain would form with these very strong biofilms, but only in aerobic conditions. So that's what I thought. More questions? We still have time. I thank you because you guys finished in good time, so we can have a good conversation.
Okay.
Well, I guess we don't have any more questions then. I thank you both and congratulations for the work.
Thank you. Thank you so much.
And we're going to have a short break. We can have some cake, coffee, and back again at 2 p.m. Thank you.
Okay, have a nice day.
Yeah, have a nice day.
Have a nice evening to you.
Thank you.
Yeah, thank you.
Yeah.
Comment ils nous ont
but my audio is okay, I guess. People online can hear me.
Hi, welcome.
Let's see.
Yes.
Hello.
Hi, welcome.
Hi, now you can hear me. Good afternoon.
Good afternoon. Hi. Hi, everybody.
Welcome, Virginia and everybody else, to our afternoon section. We're going to continue now with a presentation from Professor Virginia Alves from University of Goiás. So thank you very much, Virginia, for participating in this winter school. I'll just briefly present the bio of Virginia, and then she's going to start. So Virginia holds a degree in pharmacy, a qualification in biochemistry, specialization in microbiology and clinical cytology from Federal University of Goiás, a master's degree in pharmaceutical sciences, and a doctorate in biosciences applied to pharmacy from the University of São Paulo. She completed a 7-month sandwich doctorate internship at the Technical University of Denmark, also undertook a postdoctoral internship at the Canadian Institute for Food Safety, University of Guelph, and she has been a professor at the Federal University of Goiás since 2012, and she is a member of 2 graduate programs at that university. Her main areas of expertise are microbiological analysis of food, microbial ecology in food, and food safety. She is currently the vice coordinator of the graduate program on pharmaceutical innovation and the coordinator of food microbiology area of the Brazilian Society of Microbiology. We actually work together in the Brazilian Society of Microbiology since 2020? I don't know.
This is the third year. I think this is the third year. Yes.
Third year, so it's since 2021.
Yeah.
I've been involved with that coordination before with Nero, so it's been a while. So Virginia, thank you so much.
I'm younger, Wellington.
What?
I am younger than
Yes,
you
of
and
course. So thank you very much for participating in the winter school, and I give the presentation to you.
Thank you very much, Wellington. I will start sharing my presentation with you, and then I will not see you. Is it okay? Can you see my presentation? I don't hear you, Wellington.
Me. Can you guys put me back, please? Virginia, yes, we can hear you.
Okay, so I will.
And your presentation is not in full screen. You need to put in full screen, please.
How about now? Is it okay now?
It's still not in full screen.
Okay, just one more tentative. And now?
Yes.
Okay, thank you. So first of all, thank you for this invitation, Slavi and Wellington. It's been a few years, I have to say that it's been a few years since my last presentation in English. So my English is pretty rusty now, and I would like to apologize in advance for the mistakes I will commit during this presentation, but it happens. So I will talk today about honeybees' beneficial microbiota. And I have to start saying that I became interested in studying the microbiota of bees recently, about 2-3 years ago. But since then, this word, especially the stingless bees word, really captivated me. And today it's one of the main lines of research in our lab. So let's talk about bees. Regarding bees' importance, it's undeniable that they are crucial for maintaining the balance and health of the environment. And actually, it has been suggested that bees can actively contribute to achieve at least 15 of the 17 sustainable development goals proposed by the World Health Organization. And I'd like to highlight 3 of these SDGs now: food security, good health and well-being, and also life on land. Food security. The bees are essential for pollination services. Actually, they are among the main pollinations in the world, and with these pollination services, they can increase the productivity of several essential crops. Also, bees provide safe and affordable medicinal sources, as honey and fermented pollen, which is also known as bee bread. I will talk about bee bread further. And these products have been used in traditional and also in modern medicine to treat several, to complement the treatment of several non-infectious diseases, since these products are rich in bioactive molecules and bioactive substances such as antioxidants and antimicrobials. And also, the pollination services are important to maintain biodiversity, and the plants are important to improve and maintain air quality. So it has been very discussed in recent times, in the last years, about the importance of bees to the life on land. Actually, in the last years, in recent years, bees have faced several challenges to their survival, so that more and more studies are actually needed to contribute to bees' health and maintenance. And here I have a beautiful and colorful picture of different specimens of bees. We have now, we have about 2,000 species of bees, 2,000 different species of bees, and they have behavioral habitats which are different, morphologies that are different. And in this picture, I can show you 5 specimens of different bees. And I'm proud to say that all the pictures I will show you during this presentation are from my research group. And we have many beautiful pictures from bees and bees world. And in this picture, we have 5 specimens of bees, letters A, C, D, and E. F, actually, letters A, C, D, and F are from stingless bees. Here, letter B, I have a male, solitary male from the genus Chilocopa, which is also important for pollination. And here we have the Apis mellifera, which is the most studied and most known species of bee species. And this bee is largely known also by the products it produces. So Apis mellifera is also known as honeybee, Western honeybee, and European honeybee. It is the major species kept by beekeepers, first because of their importance for pollination of crops and other plants, but also because they can produce several products that are important for humans, such as honey, the fermented pollen, which is also called bee pollen or bee bread, and it is used to feed the hives, beeswax, propolis, royal jelly, And these products, they are important not only because they have nutritional characteristics or therapeutic properties, but they are also used in other areas like textile industry and the resin industry and others. Although these bees are largely cultivated, there are many people working with Apis mellifera around the world. These bees required specialized management due to their aggressiveness and also because they have a sting. So for working with Apis mellifera, you have to be prepared. Regarding their natural microbiota, as for all the other bee species, the honeybee has a natural microbiota and is associated to the environment where they live and to the social contact between bees. And this natural microbiota is associated with several benefits to the roast. So here I have this picture. I like very much this diagram. And here it shows this bee, the Apis mellifera, and it highlights here the main core gut bacteria that are present in their guts. And it's constituted by 2 gram-negative species, which are Snodgrassella alvi and Giamella apicola, and 3 gram-positive groups of bacteria. The Lactobacillus melliferae or Lactobacillus mellis group that previously was called firm for clade. There are several manuscripts with that still says it's firm for clade, but it's now the correct name now is Lactobacillus melliferae group. The group previously known as firm 5 clade, which is now the Lactobacillus melliventris clade, and the Bifidobacterium asteroids related species cluster. So all these groups are here, and here I have this Bombilactobacillus, which is from this Lactobacillus melliferae group. These bacteria, they are very important to their host because they can help with they are associated with the protection against pathogens in all stages of bee development. We can see that they are present since the beginning of bee development. They are introduced in the larvae and they are important to protect the bees against pathogens. And they are also related to digestion, detoxification process, growth, development of bees, immune stimulation, fermentation of food reserves. So they are crucial for the healthy development of these bees. And I also highlighted here this Apilactobacillus concave group, which although it's not from the main core gut bacteria, it is largely associated with bee products such as honey, fermented pollen. And this bacteria, we will talk a little bit more about this species a little bit later, because it's a really very interesting bacteria from bees. Regarding the stingless bees, there is a great variety of species. There are around 600 species that are currently known, And about 250 of these species are found here in Brazil and also in other tropical areas, but Brazil is the major country in the variety of stingless bees species. These bees have great potential for domestication as they are usually less aggressive than the ones belonging to the genus Apis, and they can produce valuable products to commercialization. The stingless bees processes honey in a different way if I compare with the honey produced by Apis. So the bees collected the nectar, mix it with salivary enzymes, and deposit it in these small pots that are called honey pots, where it undergoes a dehumidification process and where the microbial fermentation takes place. One important characteristic of stingless bees is that the product from stingless bees usually has a higher moisture content and also higher water activity. And this is what allows for this microbial fermentation in the honey pots. Besides that, these products from stingless bees, they have high added value due to their unique sensory characteristics, such as unique flavors, aromas, colors. There is a great variety of colors depending on the producing species. And these products are also associated with functional properties. For example, some stingless bees can produce honey which of phenolic compounds that is higher than all the other honeys produced by different species such as Apis. So, because there is this great variety of stingless bees species, it is quite difficult to define identity and regulatory standards for the stingless bees honey and other bee derivatives, other inputs. And they are also a broad field to be explored regarding their microbial diversity and the source of beneficial microbes and bioactive compounds that can be extracted from these products. Talking about the stingless bees microbiome, it seems to came from the same ancestral as the Apis genus, but nowadays we know that it is really dependent on particular characteristics of the bee species. But most stingless bees, most the studied stingless bees have Proteobacteria, Firmicutes, and Actinobacteria as the main bacterial phyla. However, there is an enormous variation amongst the dominant genera across and intra-SB species. So, although I can find the same core of bacteria, the amount of each species can vary, not only intra-species, but also in the same species, depending on the habitat, for example. But The main symbiotic bacteria associated with their microbiome are Bacillus, Streptomyces, and lactic acid bacteria. The Bacillus group is a very important group, and this gender has been studied for a variety of biotechnological applications, as the Bacillus species can produce a myriad of metabolites that have diverse potential for use in agro-industrial sectors, such as biopesticides, proteolytic enzymes, enzymes that can disrupt bacterial biofilms, enzymes that can be used in textile industry, that can be used in detergents and other diverse possibilities. And it's also interesting to note that Bacillus can be found both in bees' gut and also in bees' products. And these bacteria are associated with intestinal health of bees and digestion of carbohydrates and proteins. So they are important to the health of the bees where they are found. And also they have antimicrobial activity as they produce antimicrobial metabolites. And another interesting characteristic of the genus Bacillus is that they are spore former genus. So they can survive in harsh environments. And this makes this genus very interesting to application as probiotics, not only for bees, but also for humans, as they can survive to harsh environments, as the gastrointestinal tract of humans, where the bacteria, the probiotics, can face several challenges. Other group of bacteria associated with the SB microbiota is the group of the genus Streptomyces. These bacteria, the Streptomyces, produce several secondary metabolites of great commercial interest. They have been widely studied due to their potential to produce vitamins, pigments, antimicrobial substances, enzymes, herbicides, and they have been tested also to inhibit undesirable microorganisms, and they are associated with activity against, for example, Penibacillus larvae, Melissococcus plutoneus, which are very important bee pathogens, but they also can inhibit many human pathogens. But the last but not least important group of bacteria here is the group of lactic acid bacteria that has been discussed before this presentation and will also be discussed after my presentation. But there is a characteristic in the group of lactic acid bacteria that is of interest when we study the stingless bees, and this group has been described just a couple of years ago, not just a couple of years, but a few years ago, and this group is the group of fruitophilic lab, and we will talk a bit more about this fruitophilic lab a bit ahead. So, just talking about the lab, these bacteria are largely studied in the food industry as starters, as bioprotective cultures, As well, they are employed to improve nutritional and sensorial properties of foods. Also, labs are a leading source of probiotics. And regarding the bees, the lab are very important for the bees' health because they contribute to the degradation of vegetal polymers, carbohydrates, especially pectin, celluloses, and others. Also, they spontaneously participate on fermentations products that will give rise to bee products. And also, they are very important because they can provide protection against pathogens. They can directly protect or indirectly protect the bees against pathogens by the production of antimicrobial metabolites such as bacteriocins, and indirectly by reducing the pH of the environment where they are found. They also protect against pathogens due to microbial competition and stimulation of the immune system. But the group of F lab that has, which is a group that have been described more recently, it's very interesting because this group have an unconventional growth pattern, as these bacteria are found in environments that are rich in fructose. The F-lab species include species that do not grow in glucose environments, or they grow late, but they can develop very well in environments that contain fructoses. They are easily found in fructose-rich environments such as honey and fruit surface, and in the guts of insects that have fructose-based diets. This bacteria seems to have suffered a regressive evolution process, as they lack or they present in reduced amounts some genes. And another interesting characteristic of this bacteria is that they have molecular defense mechanisms that guarantee their survival in the presence of various plant secondary metabolites, such as phenolic compounds. Some bacteria of the Lactobacillus concae and some Fructobacillus, they have been described to they convert phenolic compounds in phenolic acids derivatives that have higher biological activity than the previous compound. So this is really interesting. Another characteristic that makes the Fructophilic lab interesting for use in the food industry is their capacity of reduce fructose content in foods, as they are able to reduce fructose to mannitol, which is a low-calorie sweetener. Also, these bacteria have demonstrated potential for use as bioprotective cultures, as they can inhibit several other undesirable microorganisms, including yeasts, molds, and many species of bacteria. I like this photo very much, and here we have an specimen a bee species named Physiomielita languida, that in Portuguese is named, if I'm not wrong, it's named Mocinha Preta or Little Black Girl. And I like this picture because it shows in the legs of the bee some this content of resin that is collected from plants and is used by bees to construct the honey pots. But I like this picture because I think it's very beautiful. So here I have a list of bacteria that are associated with bee goods and products. They are the same as described for Apis mellifera, but I destack here that most of them are F-lab, although there are some labs, but I have many F-lab here. And now we are studying the probiotic potential of F-lab. We don't have now the answer for the role of F-lab in the human microbiota, but it's assumed that individuals that consume a high fructose intake can have these microorganisms in their microbiota. So they might contribute to the digestion of fructose. Also, they are interesting because they can digest FOD and APS, which are groups of fermentable oligosaccharides, disaccharides, monosaccharides, and polyols that are poorly absorbed in our small intestine and can cause discomfort, can cause intestinal discomfort. So these bacteria have potential to be applied to manage disorders of the gastrointestinal tract. Also, it has been demonstrated that some supernatants of F-lab cultures can inhibit undesirable microorganisms, including multi-drug resistant strains. And as I said a little bit before, this Apilactobacillus concae is the species of F-lab that has been more studied. So we have some information about this species. For example, it has been observed that it increases the IgA content in saliva. It can diminish the colonization by Bacteroides fragilis. It can diminish the formation of biofilms by Pseudomonas aeruginosa. And the first bacteriocin produced by an F-lab was described is produced by this bacteria, and it's called Concassin A, and it is active against bee and human pathogens, and also this species has been demonstrated to produce beneficial enzymes that could be of importance for human health. If you want to know a bit more about this bacteria, you can check on these manuscripts that we contributed to. Associated bacterial microbiota of honey and related products from stingless bees as novel sources of bioactive compounds for biotechnological applications, and these bee-associated beneficial microbes, importance for bees and for humans. Both were published this year, and they are very informative. But now I'd like to have the opportunity to present for you some of the results of our research, the research that we are conducting in our labs regarding to the microbiota of stingless bees. This study here is a study from one of my students, Luisa, who is doing her PhD now, and she is working with this species named Scaptotrigona postica, or in Portuguese it's called Abelha mandaguari, bee mandaguari. And here I have a module containing the bees' nest, the bees' hives, and some bees were collected, desiccated to extract their crops and intestines. And also here I have the honey pot of this species, and the honey, not the male, the honey was collected and placed in bottles to fermentate, and it was protected from light. And Luisa have made the follow-up of the microbial populations in this honey. And she observed that the microbial populations decreased as time was passing. So the day of the collection, after 30 days and after 60 days, there was a decrease in microbial populations. Also, some of these, but I think she isolated around 400 bacteria, 400 isolates, and she tested these isolates against undesirable bacteria, both spoilage and pathogens, and also bee pathogens. And here we can see some of the inhibition allus produced against E. coli, Listeria monocytogenes, and Penibacillus larvae, which is a bee pathogen. Later, she is, after isolation around 300 bacteria, the number of bacteria was reduced to around 40 or 45, something like that. And these bacteria were identified by Malditoff, and all of them were Leuconostoc mesenteroides, Fructobacillus frutosum, Lactiplantibacillus plantarum, or Apilactobacillus concae. Some of them could not be identified by Maldi, but here I have some plates showing the proteolytic activity in PCA containing 2% of milk of this bacteria, and we can note that some of them had this most of them actually had proteolytic activity, and I could associate their numbers with some species here. I don't know, Luisa has the collection and she's still studying this bacteria. One of her studies has demonstrated that some of these bacteria may have potential to be studied, aiming to apply them as probiotic, as this strain, Lactiplantibacillus plantarum J4, demonstrated tolerance to diverse pHs. We can notice here that she accompanied the bacteria for 180 minutes, and it is noted that their amount did not significantly diminishes. Actually, it remained in their initial level or even growth, depending on the pH. This Lactobacillus plantarum was isolated from this little bee, which is called Abelha jataí in Portuguese, jataí bee in Portuguese, and this is the entrance nest of a jataí. And also, we are very excited about the last results we had, but we did not yet evaluate it deeply. And I have here the metabarcoding analysis of some bee products and also from crops and intestines of these species. I will try to explain here what we have so far. In this column, we have the collected honey in the day of collection. In the second column, we have the honey after 30 days of fermentation. And here in the third, in the third column, we have the honey after 60 days of fermentation. And this column, this last column here, is representative of crops and intestines of this bee species at the sampling days. So this is the bees microbiome, and here we have the results achieved with the honey. We did not have yet time to identify the specific genus, the specific species that were present in great numbers, but it's interesting to note that most of the ASV here belongs to Lactobacillus genus. Also, we have here the honey from the Jataí, the Tetaganisca angustula bee, but just at the sampling day, which also has representative of the genus Leuconostoc here. But it was possible to see that we have Bifidobacterium here. This is from the bee, which it corresponds to what the literature has shown us. But to our knowledge, this will be the first description of this bee species microbiota. And we will go deep in these results to make a better analysis to check what is here. There are lots of colors here. I could not just associate with this genus here, but I think this might bring some very interesting results. We also are studying another bee species, and we sent it to metabarcoding analysis just last week, but I think we will have some very interesting results to discuss and to publish about these bees microbiotas. So just to finish here, I would like to thank my team, our team. Here I have Professor Pedro, who is the responsible for the bees. He is the responsible for the Meliponary, where these bees and these honey samples were collected. Tatiana is another professor who works with natural products, and she loves pictures. We are preparing a book with beautiful pictures for soon. Professor Elaine, who is not only a great friend, but also a great collaborator of our studies for a very long time. And my students, Gisana, Luisa, Ingrid, and Ruama, who are actually the responsible for these results we have so far. I also would like to acknowledge the CAPES, FAPEG, CNPq, and Confederação Brasileira de Apicultura e Meliponicultura, and AP Goiás, who have also contributed to these studies. I hope it was possible to understand me. And if you have any question, please, you can do it now.
Thank you, Virginia. Can you hear me?
Yes.
Okay, great. Thank you. We have some comments and questions in the chat, so I'll forward them to you before passing here at the audience. Raul Rosa says that he's passionate about native Brazilian bees. He has 3 different species at home and has already identified 7 different ones on campus. So it's something he's gonna be studying in the future. Icaro asks, today we know that pesticides can promote the emergence of antibiotic resistance in bacteria through mechanisms of cross-resistance. How do pesticides and other agricultural practices affect the bee microbiota, and what are the potential consequences for bee populations?
Icaro, this is a very good question, and actually what we have noticed so far is that the pesticides that are used, for example, for combat, for exterminate the ants, they are much more effective against bees than against other insects. So they do not affect only the bee microbiota, but the bees themselves. These pesticides can kill bees much easier than they kill another insects. So it's important to study the bees to maybe we can find some bacteria that can positively influence bee health status, a probiotic that can help in this way. But so far, what we know is that they are very deleterious to bees.
Yeah. So Raul didn't understand where the samples were collected from. So he wants to know if they were collected from the honey itself, or the hive entrance, or from some other place.
Raul, actually, at UFG, at the Federal University of Goiás, there is a meliponary there, and Professor Pedro is the one that takes care of the bees. We collected the modules, the nests, and we brought them into the lab, and we aseptically collected the honey on our lab. And also the desiccation of bees were performed in our lab. Did I answer it, Wellington?
I think so, yeah. Luna asks, did you study the difference of the honey and gut bees microbiota according to the type of flowers?
Not yet.
Yeah.
Not yet.
And again, what about microbial diversity throughout the year? Different seasons, honey has different moisture content, and bees visit different flowers. Any evidence that this has any impact? Or is the microbial diversity exclusive species dependent?
No, the microbial diversity does not depend exclusively on the species. Actually, we have our studies have started just 2 years ago. But now it is interesting that you did this question, because we are studying one bee species that was fed by the beekeeper, and now we will collect in August, because the seasonality, we will collect samples from the same hives with but now the bees will will naturally feed themselves. They won't have help. So we will compare just these 2 situations. But probably the seasonality has a huge impact, maybe not a huge impact, but it can impact the microbiome, but we don't know how much. I think that the main core species will be present at any situations in the same beehive.
Yeah. One last question from the chat. Very interesting presentation. Have you considered studying the potential of using these types of honey for production of mead or other fermented beverages?
We did not have time yet. We are not studying it for foods. We just it as well. I don't know if it's very difficult to have money to afford these researches, but we are just starting them. But we are looking for collaborations that in other sectors, maybe in the near future, but not yet.
Yeah, sure. Any questions from here, the audience? Lots of interest for your work, Virginia. Congratulations.
This is great. I would like to share my email. I will put just a minute here.
You can add in the chat if you want.
Yes, okay. I'm not in the chat. I'm just.
Or you send it to my email and I'll put it in the chat.
Okay.
You send to my Zap and I'll put in the chat. Or Slavi, yeah.
Yeah. Please share my email with the audience and with the people in the chat, and it will be a pleasure to talk about this captivating world.
You know, yeah, many people are passionate about bees and honey and all of that. So congrats.
Thank you.
Thank you very much. And now we have a short break until the next talk at 3 p.m. Take care. Bye-bye.
Bye-bye. Thank you very much. Bye-bye.
Hello, everyone. Welcome back. Hi, Rachel. Nice seeing you.
Likewise, hello.
Thank you very much for agreeing to participate in our winter school. It's a great pleasure having you. Yeah, it's summer. It's California, I suppose, so it's great.
Yes, beautiful day
Rachel,
here.
Yeah, so I had the pleasure of watching a talk from Rachel at Harvard. I think it was 2019, and it was a great talk. And I was like, wow, I hope I can invite Rachel sometime to talk for us in some event. And here she is. So I'm very grateful that you accepted my invitation to be part of this winter school. So thank you so much. Rachel is going to be talking about fermented foods as tractable microbial ecosystems. Rachel Dutton is an associate professor in the Division of Biological Sciences at UC, University of California at San Diego. She's a microbiologist and currently an entrepreneur in residence at Astra Institute. Over the past 14 years, first as a Bauer Fellow at Harvard, then professor at UC San Diego, then scientist at Arcadia Science, she has led the development of cheese microbiomes as useful models to study interkingdom interactions between bacteria, fungi, and phage. Dr. Dutton has been named an NIH Director's New Innovator, a Packard Fellow, and a Pew Scholar, and her work has been featured in the New York Times, Wired, NPR, and Netflix. So we really look forward to hearing what you have to say for us today, Rachel. So the floor is yours.
Great, thank you. And let me know if there are any issues, but looks good on my end. So thank you so much for the invitation. I wish I could be there in person, but we are right in the middle of summer here in Berkeley, California. So it's nice to be here too. So I'm going to tell you about the work that I've done over, for over a decade now, thinking about cheese and fermented foods. And initially, this work was done in my academic labs. 2 years ago, I moved from UCSD to Arcadia, and then just a few months ago into this entrepreneurship position at the Astera Institute. So I'm going to tell you just a little bit about what I'm thinking about in this new position that I have. But where I got started in working on fermented foods was this question that sort of kept coming up in my head when I was an E. coli biologist. So my PhD was in E. coli genetics. E. coli is amazing, extremely powerful organism to understand biology, but I kept thinking about all the other organisms that we were missing and all the biology that we were probably missing, because in most cases as microbiologists, we study organisms in isolation. And so I wanted to find a system where we could actually sort of work in both directions, from microbial ecology in more complex systems, bring those organisms into the lab and start to understand mechanistically what's happening. And then that mechanistic understanding will hopefully shed light on the environmental behaviors and biology that's happening. And this is what really brought me to fermented foods, and in particular in cheese. And so as I was looking for these systems that were simple enough to be able to bring them into the lab and dissect them, so at the time, people were starting to study the gut microbiome and ocean microbiomes, which are incredibly complex. But fermented foods, as you know, are relatively simple. And cheese really caught my eye because it had a broader phylogenetic diversity than many other fermented foods. So it felt to me like an environment where there was really a lot to discover still. And so what I decided to focus on are these microbial communities that form on the surface of cheese as they age. So this is the rind biofilm that forms on the top of the cheese. These are 3 different types of cheese made in different ways by cheesemakers, aged in different ways, and they grow different types of microbial communities. And what we knew at the time was that they were relatively simple. And then what we went on to show was that we could actually map out the diversity of species in these cheeses. So we went around and did field work, which for us meant traveling to different countries and collecting cheese samples and scraping the rinds and sequencing the DNA. We then took some of those samples and used them to deconstruct the communities. So we then showed that of all the species that we saw of bacteria and fungi in the system, we could actually culture them in isolation in the lab, which is quite different than most environmental microbiomes where it can be challenging to cultivate organisms in the lab. And then we used this collection then to start to rebuild communities in the lab. And that allowed us to sort of mix communities of any combination of species that we wanted and try to understand, okay, how are things different when these organisms are growing by themselves versus when they're growing together as communities. And that took probably the first 5 years of my lab's research to kind of just get the system set up and show that this is an ecosystem that we can actually bring into the lab and study it. And so the next about 10 years of the research that we did was really then trying to see, like, how do we leverage this power to bring a community in the lab to understand biology? So we use a combination of what I think of as top-down approaches, so things like sequencing based approaches, where we're going out, collecting samples, sequencing them, trying to look at patterns that we can then, and build hypotheses that we can then test in vitro in the lab. And then cases where we would do experiments in vitro in our cheese environments in the lab, and then we could, using genetics and in vitro community manipulation, learn about the mechanisms that were acting here and use that to infer what was happening in the environment. Okay, so I'm going to talk about sort of 2 main areas of research that we embarked on in my lab. So the first was going from this bottom-up approach. So trying to see, now that we have these organisms in culture, can we do genetics and can we learn about the mechanisms that are driving microbial interactions in cheese? And the way that we did this was we adapted a transposon sequencing approach that was developed here in Berkeley by Adam Arkin and Adam Deutschbauer's labs, where they have randomly barcoded transposons. And what we did is we, instead of directly mutagenizing one of the species from a cheese, we developed this approach where we use an E. coli transposon mutant library as a sensor. So let me tell you what this looks like. And this is work that was led in my former lab by really talented scientists, Manon Moran and Emily Weiss. So this is how this experiment worked. And we use this across many different types of experiments, but this is the general idea. So you have an inoculum, and so this is our E. coli transposon mutant library. So every cell, in theory, in this population has a mutation in a different part of its genome, and every mutation is linked to a unique barcode. So what we can do to track the abundance of different mutants in the population is just sequence their barcodes, and we look at the abundance of these different mutants in our inoculum, and then we take that inoculum and grow it just by itself on cheese. Now, some of the mutations are in genes that maybe are required for growing on cheese, so these mutants die, and which means that those barcodes will then drop out of the population. And then we can compare that next to the growth of the inoculum on cheese, but in the presence of a microbial community. And this perhaps will have some of the same mutations that have an effect versus others. And so we now then sequence the barcodes in this experiment. And really the power comes in comparing across all of these different conditions. So for example, we see in the inoculum, you have a mutation present. However, in the cheese environment, this mutant can't grow, so it drops out. in the community, it also can't grow. So this is considered a core requirement of just growing on cheese. Now, this one is interesting. This is a mutation that we have in the library. It can grow fine on cheese, but in the presence of the community, now this mutant dies. And the other scenario is we have a mutant that's present. It doesn't grow by cheese. It doesn't grow on cheese by itself. However, in the presence of the community, now it can grow. So there's something about the interactions there that are alleviating the defect of this mutation. So we applied this in a bunch of different scenarios, but the one I'm going to talk about today is trying to understand the role that fungi play in microbiomes. So fungi are often understudied in the context of microbiomes. Most of the technology that we see being applied to metagenomics and other types of sequencing is really focused on bacterial populations. However, cheese provided this really nice opportunity to look at fungi because we had a decent amount of diversity and abundance of fungi in the system. And so my first grad student at UC San Diego decided to take this on as her thesis work. And she screened a panel of cheese-associated fungi against this E. coli interaction sensor library. And I don't expect you to be able to interpret all of this, this chart here, but the points, I have a couple of main points I want to make. One is that fungi have a really diverse impact on bacterial fitness in this environment. So this is looking on the x-axis at the growth defect when the E. coli library is grown by itself on cheese. So there are mutations here that, you know, if they had no effect, they would be up here around the 0 intersection, but further left means they have a stronger growth effect. However, then on the y-axis is now the average fitness with a fungal partner. So, for example, these genes in pink here have moved up in fitness in the presence of fungi compared to where they were in their fitness growth alone. So what that's saying is that the fungi are actually improving the growth of these mutants. And these mutants are iron uptake mutants. So let's look at that in one other example, and then I'll tell you a little bit more about iron in cheese. So what this is showing is these 6 genes here that are all involved in iron and siderophore uptake. So siderophores are small molecules that chelate iron from the environment. Now, if any of these mutants didn't have an effect, they would grow here at the top of the chart. In the white is their growth by themselves. So this is just E. coli growing on cheese. However, in the presence of any of the fungi we tested, now these mutants can restore some of their growth defects. Okay, so it's just saying that there's something about the fungi being present that's making it so that that E. coli doesn't need its siderophore uptake system anymore. So what do we think is happening? So through a number of other experiments, which I'm not going to go into details, but we published the paper several years ago, what we think is happening is that actually these bacteria in E. coli can actually steal fungal siderophores. So in many environments, and this is not in any way limited to cheese, iron is a very limited nutrient. And in the presence of oxygen, it becomes oxidized and is insoluble. So bacteria spend a lot of energy making small molecules called siderophores. They use non-ribosomal biosynthetic machinery pathways to produce complex small molecules. This is not doing the molecule justice here. They produce export systems to pump these out of the cell. These go into the environment and chelate iron, so they can solubilize it. Then they have uptake systems. In this case, this is the E. coli FEP system, which is where we saw all of those mutants, that can bring these iron-bound siderophores back into the cell so that E. coli can have enough iron to grow. So in this library, what we saw is that if E. coli can't take up iron siderophores, it can't grow on cheese when it's by itself. But in the presence of fungi, now it can magically. grow in cheese. So what we think is happening is the fungi are producing their own siderophores because they face the same challenge of needing iron. And it turns out that E. coli has a fungal siderophore uptake system that's been known for a long time. It's called the Fuse system. And what we think is happening is that E. coli, in the absence of its own siderophore uptake system, but in the presence of fungal siderophores, it can actually use its fungal uptake system to steal fungal siderophores from the cheese fungi. And what's interesting is that all of the fungi that we tested, including isolates not from cheese, from fungi from skin and soil, have a similar effect on E. coli. And so what we think this says is that this is maybe a widespread phenomenon where bacteria are basically stealing the siderophores from fungi in their environment. Okay, so that this transposon library using E. coli told us a lot about something that is happening potentially in the environment where fungi are producing siderophores that are accessible to bacteria. Another relatively understudied part of microbial communities are actually viruses. And so we wanted to start to think about what are the role of phage in the context of communities. So often when phage are studied, they're studied in the context of their single host and not thinking about, okay, what happens when the phage and the host are in an actual community. And so this is work that was led in my group by Tara Spencer and Gary Huesler. So first we had to see, okay, can we actually find phage in cheese rinds? Phage, anywhere there are bacteria, they're going to be phage, but we had to go through and purify some. So we took a brie type cheese, and we took a bacteria that we had isolated from that cheese, Hafnia alveoli, and we used it as a host to try and identify phage. So you can see here a dilution series of extract from a cheese rind where you can now at the end start to see individual plaque forming units. So we have isolated a phage that infects this cheese bacterium. And so we wanted to ask, okay, what does this phage do in the context of community? So in this case, we actually turned to making a library in a cheese species. So we use the genetic tools for the library construction on Hafnia to make a large Hafnia mutant library. And then we grew this library in different combinations. So these are the different in vitro community combinations here. So growing it just by itself, the bacteria plus the phage, the bacteria plus the community, and the bacteria with the phage and the community. And then with each of those, went through and looked at what was happening. And what we found was actually pretty surprising. What we found was that the fungi seem like they're actually suppressing the ability of Hafnia to become resistant to phage. So all of the conditions with no phage are in these solid lines. So what you can see is that when Hafnia is growing by itself or with either of the fungi or with both of the fungi together in this community, it can basically grow just fine, as expected. However, in the presence of phage, now this phage is called TS33. Hafnia, when it's growing by itself in gray here, in the presence of this phage, initially starts to decline in population numbers. However, it bounces back. And then you can see sort of some decline, and presumably after this point, there would be some resistance and it would bounce back again. So you see some amount of phage resistance coming up. However, in the presence of any of the fungi or the two fungi together, it reaches the same point of population loss, but then it doesn't recover. So eventually in these conditions, Hafnia just goes extinct. So it's not able to become resistant to this phage. So what is going on here? Well, the mutations that we get in the Hafnia that are resistant to phage all map to cell envelope pathways. So these are mutations in the biosynthesis of what we presume is the O antigen of the lipopolysaccharide outer membrane of Hafnia. So Hafnia is a gram-negative bacterium. So in wild type Hafnia, it is very susceptible to phage. So you get lots of phage growing in the presence of a wild type Hafnia. However, when you get rid of any of these O antigen biosynthesis genes, now the bacteria become resistant to the phage to different levels. However, when we looked at the growth of these different mutants with fungi, what we saw is that it actually slightly increases the susceptibility of Hafnia to fungi. So wild type Hafnia can grow just fine with fungi. However, any of these mutants now have a decrease in growth in the presence of fungi. So what we think is happening is that these mutations that normally would give phage resistance to Hafnia, which would allow Hafnia to escape predation by the phage, in the context of a community, it can't grow because it actually increases its susceptibility to fungi. So what we think this means is that the bacterial cell envelope is actually a focal point for this evolutionary trade-off. So if you can imagine, gram-negative bacteria are protected by this outer membrane layer. However, if you have a mutation, let's say because you've created this mutant library where you have random knockouts, some of those knockouts will have mutations in this outer membrane biosynthesis pathway. So now you no longer have this outer membrane. Well, that could be good in some cases if the phage itself is using that outer membrane as a receptor. So you can actually escape predation by the phage. However, if you now grow up, but you're in the presence of fungi, now you no longer have this protective layer on the outside of you. So you might be more susceptible to otherwise sub-inhibitory concentrations of antimicrobials produced by fungi. Okay, so I'm gonna move on to more of our top-down. I don't know if you want me to pause and take a moment for questions here, or should I just keep going and take questions at the end? I'm happy to do either.
I think you can keep going. That's
okay.
Great.
Okay, great.
Yeah.
Okay, let me take a sip of my tea. Okay. So far what I've showed you is how we can do this sort of bottom-up approaches using genetics to really understand the mechanistic basis of some of the interactions in biology that are happening in these communities. What I wanted to show you next is how we have applied some of the sort of top-down sequencing-based approaches to also shed light on some of the biology. So the area here that I'm going to focus on, I mean, top-down approaches can shed light on many different areas of biology. The thing that I found most interesting in the system is this idea that the microbes in cheese and other fermented foods probably need to adapt really quickly to live in this environment. They're coming from different places. So for cheese making, they would be coming from the udders of the animals, the barn environment, the cheesemakers and other places, and then find themselves in milk. In other fermented foods, this would be the same, where let's say in fermented vegetables, they would be associated with the plants themselves, and now being put into an environment where many of the biotic and abiotic conditions are quite different than what they had evolved to live in. So, for example, they may find themselves around other neighbors that they haven't seen before. And some of those neighbors may be doing things like producing antimicrobials or siderophores. And then the abiotic environment is also very different in the fermented food. So we have often low pH. In the case of cheese, we have the presence of lactose, which is a different carbon source. And so I was really interested in this idea that what happens to the microbes when they are growing in these foods? How do they adapt? And could we use these sequencing-based approaches to detect what's happening? Now, when microbes face themselves with a new type of environment, one of the most powerful ways for them to rapidly adapt is through horizontal gene transfer. So this is where, pieces of DNA from one organism can be transferred to new organisms, either through phage, through conjugation, or through competence pathways. This is mostly studied in the context of antimicrobial resistance for nosocomial pathogens in hospitals, but this same process is happening everywhere on the planet with microbes every day. So we wanted to know, how common is horizontal gene transfer in cheese? And this is a project that was led by a former postdoc in the lab, Kevin Bonham. And what he did was take the genomes of this collection of about 165 different cheese-associated microbes that had been isolated and sequenced and were available in public databases. And the analysis for doing this was basically looking for regions of a bacterial genome that had an unexpectedly high similarity to a distantly related species. So you'd expect lots of similarity within, let's say, the Bacteroides, but you might not expect there to be a lot of similarity between the Bacteroides and Proteobacteria. So we were looking for stretches of the genome where there was almost 100% identity over at least 500 base pairs between these pairs of genomes. So he did pairwise comparisons for every single genome here. And what he found was that there is actually really extensive horizontal gene transfer in cheese. And so each of these lines is connecting two species where we had evidence of these regions of high, unexpectedly high similarity between pairs. The thicker the line is, more DNA shared, but you can basically see exchanges throughout this tree. And in total, these regions encoded over 4,000 genes. So this is an extremely extensive network of horizontal gene transfer just within cheese-associated bacteria. And one of the things that we kept finding over and over again in this data set were genes, of the 4,000 genes that were encoded in these horizontally transferred regions, were genes involved in siderophore uptake. Perhaps not surprising given what we now know about the role of siderophores in interactions in cheese. And so on multiple mobile elements, what we found are these uptake systems. So this is an integrative and conjugative element. So it's a type of mobile element that can hop into a genome at the 3' end of a tRNA, insert itself, and then excise itself and move by conjugation to other cells. And within this 50 kb element, there were a number of genes, but a common set of genes that we found across many of them were these siderophore uptake pathways. Now, what this is telling us is probably the ability to take up different types of siderophores is being transferred across different cheese bacteria. And then this may allow them to adapt to some of their new neighbors and potentially steal siderophores from their new neighbors. So after we saw this, this was sort of a static view documenting HCT across many genomes that have been isolated from bacteria from many different cheeses. We wanted to see if these evolutionary processes were things that we could actually see happening on a wheel of cheese as it's aging. And so this is a project led by former postdoc Christina Sock, and she collaborated with cheesemakers that we've worked with for a very long time, Jasper Hill Farm. They're in Vermont. They have these large underground cheese caves. And what they did was send us samples of 3 different types of cheese at across the aging period of these cheeses. So these are all wash-rind cheeses. You can see it's sort of week 1, they don't have much of a community on the surface, but by the end of their aging period, they now have these microbial biofilm rinds on the surface. So what she did was, for each of those time points, scraped the cheeses, used a combination of long read sequencing, short read sequencing, and then did isolations from these cheeses as well and sequenced isolate genomes to map out the temporal dynamics of the communities on these cheeses. And so, for example, this is basically a catalog of all the species we found on these cheeses. So across different phyla, and the time and their abundance in the different cheeses. So you can see these sort of dynamics over time. And then we asked, okay, now that we've sequenced these populations, can we actually see any transfer happening? So for this, she used this combination of short read Illumina sequencing, PacBio and nanopore sequencing. And this is to try and get broader genomic context of sequences as well as potential plasmids. And then she used Hi-C, so this chromosome capture methodology, which links plasmids with their host genome. And this is really important for detecting horizontal gene transfer because often this is happening through mobile elements like plasmids. And in a normal metagenomics pipeline, these connections are going to be lost because the cells are lysed. And so the plasmids cannot be confidently associated with their host genome. So after all this, we actually detected a small handful of potential transfers of plasmids. And so this is looking at the connections between plasmids and bacterial genomes at week 2 in one of the cheeses. And this particular node here is a plasmid that contains iron uptake genes, so siderophore uptake genes again. And at week 2, it's connected here to a Cyclobacter genomic sequence. However, on week 4, we now detect that this plasmid appears to have moved over to a different host. Now we find it associated with Pseudo-Enteromonas. So this is, I think, one hint that we can actually detect horizontal gene transfer, and is really, I think, kind of amazing that it's actually the plasmid we found it in actually contains iron uptake genes, which is something that all of our studies have been telling us is really a key aspect of this environment. Okay, so basically everything is pointing to iron-base interactions in cheese. And so, in a normal situation, the bacteria are creating it in isolation. Bacteria are creating siderophores and using them to chelate iron and bring them up. However, if you are able to acquire the import system that will allow you to take up siderophores from another bacteria, you can basically cheat. So you don't have to make them yourself. You can just take your neighbors. And this is also true if you can have a system, maybe a different system that takes up siderophores from other neighbors. Okay, so what did we learn from this combination of approaches and all this time spent studying cheese? A couple of big things. I think one of the big things that we learned is that fungi are really critical players in interspecies interactions. We know this is the case in cheese, but I think this is probably a much more widespread phenomenon. And fungi should, I think, be more research should go into thinking about the roles that fungi play in interactions in different environments. We think that iron is a major driver of interactions in evolution in this system. Again, this is true probably not just in cheese. This is something that's been observed in many different environments. And I think one of the sort of cool things from the horizontal gene transfer approach is that mapping out the horizontal gene transfer in a community actually told us about the evolutionary pressures and the strategies that microbes use to adapt. So this sort of top-down approach was incredibly complementary and sort of confirmed the bottom-up genetic approaches that we use. The other thing, which I haven't really mentioned, is that even though we were able to learn a lot about what was going on in the system, a lot of the insight that we had on these interactions was really limited to well-characterized areas of biology. And so we focused on iron and siderophores, partly because it was a very strong signal, but also because people have studied siderophores for a very long time, and we knew a lot about the basic biology. And so it allowed us to really quickly develop and test hypotheses about what was happening. But in our genetic screens, for example, over 50% of the genes that we found that seem to be important in interactions were found in genes of unknown function. And so I think this speaks to the fact that we really need a lot more work to understand what these unknown genes are and their contributions to microbial biology. Okay, so I'm going to just quickly acknowledge my former lab and collaborators and funding for this this part of the research, because I'm going to move just for the last bit here onto what I've been working on since moving over into a new role. So about 5 months ago, I moved into an entrepreneur in residence role at a nonprofit called Astera. And my goal there is it's basically a startup incubator. And so what I've been working on now is exploring the potential opportunities for leveraging fermented foods for real world solutions. So what is Astera and what is this thing that I'm doing? So Astera is a nonprofit. They have started now a couple of residency or fellowship programs where they are investing in entrepreneurs that are trying to do projects that don't really fit neatly into existing areas of academia or industry. And so what this residency provides is time to develop ideas and then start up seed funding to launch new either for-profit or nonprofit companies from this. And so given my background in fermented foods and interest in this space, what I've been working on is starting to think about where the opportunities and needs are really for translation in the fermented food space. And so I just find fermentation so incredibly fascinating. I mean, beyond just the amazing biology that you can find within these foods, as a cultural and historical phenomenon, it is absolutely incredible. So fermentation is what is known as a human universal. So it's our oldest biotechnology, and every culture has a tradition of fermentation because this is an innovation that happened before the migration of human populations into distinct areas of the globe. And it's thought now that fermentation may have actually impacted the trajectory of human evolution. So there has been work from largely a biological anthropology focus, but suggesting that fermentation may have impacted the size of our guts, so the shortening of our digestive tract, which therefore led to increased energy available to support a larger brain. There is molecular evolution evidence suggesting that fermentation may have been involved in the evolution of our preference for sour foods, which is somewhat unique in to the human species. Other vertebrates that have sour taste receptors, most have them, but they're usually an aversion to sour. And our ability to metabolize alcohol is fairly unique. And then more recently, there's been work on the connection between our immune system and fermented food metabolites. So there's immune receptor, GPCR, HCA3, where the only known molecule that binds this GPCR is a metabolite from lactic acid bacterial fermentation. And so there's beginning to be, I think, more and more evidence that there are these connections between fermented foods and human biology. And this can be seen if you sort of scan the literature across many different fermented foods, where these microbial bioactive molecules are really connecting fermented foods and human health. So very diverse impacts from mental health, metabolic health, cardiovascular health, gut health, and inflammation. And these are being controlled by the molecules that microbes in fermented foods are producing, such as peptides and secondary metabolites, different acids, exopolysaccharides, as well as enzymes. Now, these connections have been more and more recognized over the past few years and seem to play important roles in human biology, but I think we're really still at the tip of the iceberg about what's happening here. And it's fairly concerning to me, given that the industrialization of the food system, especially in the Western world, has dramatically altered our foodscape. So about 70 years ago, we really transitioned from microbial processing of foods to industrial and chemical processing, refrigeration, preservatives. And so the result of that is the diversity of fermented foods and overall intake dramatically declined in the Western diet. So, we're no longer getting many of these bioactive molecules that have been a part of the human diet for many, many years. The sort of like another consequence of this shift in our food system has also meant that it shifted scientific research priorities in food science. And so, this connection between our human health and fermented foods are really only recently being uncovered, because fermented foods had been dramatically understudied for decades, because most of the food science was going towards industrial processing of food and not focused on these traditional practices of fermentation and their potential benefits. And so this, I think, has had a really stifling effect on innovation and on translation in this space. And so what I'm working on now, I'm in the process of trying to formulate some potential ideas about this connection between fermented foods and human health. And I think it's really important because we have so many health and sustainability issues related to food. There has not been enough foundational fermented food research in academia. And so people who want to start startups in this space have a much bigger technological gap to traverse, which makes it harder to get funding and to get success in this space. And so I think it's really important that there is scientific de-risking in this space, both in the context of academia and in alternative science capacities, such as potentially at Astera, where this can be done in conjunction with open science practices and really try and open up innovation and future funding in this space. So what I will actually be doing and developing is still to be determined, but I just thought I'd give you a little preview of some of the things that I'm starting to think about. And with that, I will say thank you for your time and happy to take any questions.
Thank you so much, Rachel. It was amazing, your talk. I really enjoyed it. And I was holding a question about this Astera thing that you just presented. I was like, what is that? So I would ask you, and then you nicely showed us in part 3 of your talk what that is, which is kind of amazing and very interesting. So, and you're in California, so of course you would be involved in that. So it's expected, I guess. Let's see. Yeah, many people congratulating your talk. I don't see questions yet. I do have one though, because in the first part of your talk, you showed, in the second also, the involvement of the siderophores and all, but with the mutant, right? So fungi can improve the growth of iron uptake mutants, but how do you see this in the actual community where you have, wild types, or they are not mutants like E. coli? So how does that play out in the general community?
Yeah, so I mean, the wild type microbes of so E. coli wild type can grow just fine on cheese by itself. It doesn't need the fungi because it has its own siderophore uptake system. But I think the ability to take up fungal siderophores is something that I think could provide, certain benefits, especially, as we saw with horizontal gene transfer of some of the uptake systems. I think, for organisms that are maybe haven't grown in these environments before, it may provide them opportunities for additional sources of iron.
Yeah. Iron seems to be a very big deal in this environment.
Yes.
Earlier and also early yesterday, we talked about microbial interactions in food ecosystems, especially in cheese, and we talked a lot about microbial succession.
Mhm.
Have you saw something on that theme, and have you seen something related to iron as well? Because for me, it seems like the iron thing, at least in my mind, was something like secondary, but as you are showing here, it seems to be a major player. And when I think about fermentations, I think about cross-feeding. I think about microbial succession, because one organism produces lactic acid, the next one is going to use it and produce propionic acid, and so on and so forth. These are like major components in the milk. And the iron thing, it was kind of surprising to me to see how important it seems to be. It's just a comment, I guess.
Yeah, I think it is surprising. I think that so there's a couple things. One is that the lactic acid bacteria that are playing an important role in the initial stages of fermentation of the milk, they don't actually need iron. I think that's one of the reasons they can grow so well in milk, because it's such an iron-limited environment. They use, I think, manganese in place of iron for most of their enzymes. So it's not a big problem for them. But once you start to have these aerobic organisms trying to grow on the surface of the cheese, now they all need iron. They're a variety of different types of bacteria, and on the surface of the cheese, the iron would all be oxidized, and so you really do need siderophores to be able to maintain growth. Now, I think over the course of succession, yes, you're going to be kind of like switching different types of primary carbon sources and other nutrients that the organisms are going to be using to feed their metabolism. But if you don't have iron, you can't really do anything. So it's kind of like a baseline requirement for growth. And the other thing, in my mind, that's like a primary, and then the other things are secondary. Sure, as long as, and once you have iron, then you can figure out like what type of sugar or other carbon source you might be able to get access to.
Cool. I also find it fascinating that you could see horizontal gene transfer almost like in real time during the maturation. So it's quite amazing. So we do have a question from Alessandra Mello. She was actually a speaker earlier today. Fascinating. Thanks for the great talk. What happens to the growth of the cheese fungi when E. coli steals the fungi iron? Is the growth of the fungi affected? Yeah, that's a great question.
Yeah, great question. I don't know that we very closely looked at that, but we didn't, I think on a course level, we didn't notice any strong effects on the fungi. My guess is that they're producing enough so that it doesn't make a huge difference on their biology. But yeah, it would be interesting to think about that some more. I don't know for sure.
Cool. Thanks. Anyone from the audience here can wants to ask a question? Well, yeah, Rachel. So I don't know if I understood correctly the Astera part of your talk, but it's like an incubator for startups or great ideas. Is that it?
Yeah, so they're supporting founders who are coming from different places to develop their ideas and build and supporting that building process. But they have a very, very strong focus on open science practices. And so really trying to push the envelope of even for like for-profit startups, like how much of the data that's going into these companies can actually be made public and be shared and help lift up other areas that make it easier for more people to innovate and start companies. So it's a really interesting organization, and yeah, it's I feel extremely fortunate to be in this position to have the support that I have right now to try and build something new.
Yeah, and what they are supporting you to do is research, or you are making a product? I didn't get that part.
Yeah, they are supporting me to build something, either a nonprofit, which would be primarily focusing on research, or on a actual startup company like a biotech startup to produce a product. So I'm still in the process of figuring out what the right opportunity is for me, and what I think would be most impactful for the fermented food research and translation space.
Yeah, I see. Well, with that, we close the day, and we thank you so much, Rachel. Hope to see you in some other time, and when I visit California.
Thank you. Yes.
Thank you, and have
All
a
right.
nice day.
Okay, thank you. Bye.
Everybody for watching. We'll see you tomorrow morning at 9 a.m. Brazilian time again. Bye-bye.